All right. I'm back, and also welcome, Daffy. Great to see you. And well, yeah, I've been absent for a little bit, a few months, but I'm back, and should be back for a while. But yeah, I don't want to keep the, I guess, student waiting. So I'm going to get right into it. But yes, thank you so much for passing by, Daffy. Always appreciated. And like, yeah, we're just going to do some biochemistry, like I've done before. Hello, can you hear me?
Yes, I can.
Yes, perfect. All right, I'm gonna set up a few things here. Give me a moment.
Sure.
Turn on my camera and share. Hopefully you're seeing something.
Yep.
Okay. And then I'm gonna just do a little bit of adjustment on my side. Okay. It's been a minute since I've done this, so a little bit rusty.
Yeah, no worries.
But it should not take long.
I don't know how the weather's been there, but like all the snow melted here, and now it's starting to snow again.
Yeah, there was basically an extreme weather warning issued here of like pretty same thing. We had snow, and then it melted, and it's been raining a little bit all day here. Then I think it's expected to like freeze overnight and like
yeah, of course.
where like it's apparently supposed to be like really bad.
Yeah, that's never fun. at least so far, I mean, this area seems much milder compared to Edmonton area.
Yeah, I would imagine. Yeah, I can only imagine since I've never been there, but I can only imagine
Oh yeah,
the amount of ice and snow and extreme cold that you get there.
yeah, lots of -50 days.
Yeah, yikes. Yeah. Isn't that the point at which like they just tell you like don't stay outside for more than like 2 minutes at a time, otherwise you're.
Yeah, pretty much. That was like I walk my dogs religiously every day, and there was like I think last year maybe one stint of like 5 days where it was just like, yeah, there's no way.
Yeah, I can't like even so. At least I like to think of myself as pretty cold tolerant, as in like so like the most extreme here that we get pretty extreme. It was still like -30, -35, but like -40 with the wind chill.
Yeah.
And like I can tolerate that pretty. Like I obviously don't like it, but I can sort of tolerate. But I can only imagine
Well,
-50.
the funny thing is like honestly that is a pretty extreme cold, but then I swear the humid cold that we have here in I guess like you know mid southern Ontario, it's like it just cuts through your clothes so much more. It has like an almost colder feeling.
Oh, okay, interesting.
I just find like there's not much difference between like once you're past -10, the -10 to -30, it's like you can't really tell the difference much.
But yeah, that I definitely know that piercing cold. I don't know how windy it gets. Like I imagine it gets like just as windy, even more so in Edmonton than. But like yeah, like when it's
Yeah,
-30
not
here.
particularly.
Oh really? Oh okay. But yeah, when it's -30-ish here, and it's no wind, like I can actually tolerate it like just fine. As in like I can like probably be out in like a hoodie and I can survive for like 20 or be fine for like 20-30 minutes. But
Yeah.
when it's like wind, and it's just like amplified, like you feel like at least I feel my skin like freezing in real
Oh
time.
yeah. Oh, it just like it cools you to your bone.
Yeah, it bites. Okay, I think we're good. I think we're good. So can you see when on your end when I scroll?
Yep.
Okay, great, perfect. Then I think we're good. Okay, good to go.
Wait.
Took a little bit more time than I anticipated, but the
Oh yeah.
okay.
Nice.
Picking up where we left. So yeah, trying to finish this off as quickly as possible. Okay, so yes, signal transductions. You've seen this before. Nothing overly new except maybe the adhesin receptor and the integrins. But even then, all we're really dealing with is it. Nothing we haven't seen before in terms of we're dealing with a bunch of proteins.
Yeah.
That's about it. Just as we've been all consistently doing throughout the entire course, or all of biochemistry. Okay, so diving right in, and well, I guess to also keep it in mind, okay, that like generally speaking, for virtually all of these signal cascades, especially like the tyrosine receptor kinases and the G protein couple of receptors, our endpoint is typically like to alter gene expression, either upregulate or downregulate. Where usually for the not usually, but for many of the signaling cascades, it's like this transcription factor, as we'll see, they're sort of like showing it here with the kinase cascade. Of like it goes into the nucleus, or it's a transcription factor that goes into the nucleus, and then we upregulate or downregulate transcription.
Right.
And therefore, upregulate, downregulate the genes. Okay. And well, you probably already know this in terms of all of the different roles that they play, especially the GPCRs, as you know from previous courses, like huge class of receptors that have roles in virtually everything that you can think of, as they're showing here. So yeah, just diving right in. Yes. These secondary messengers, yeah, you have your cyclic AMP, your cyclic GMP, and your abbreviated inositol 1,4,5 triphosphate IP3 that we've seen multiple times before. And again, this is ultimately for the purposes, ultimately for the purposes of altering gene expression, but then also signaling cascade to amplify a message and exponentially amplify a message. Okay. So, getting back to the G protein coupled receptors, okay, which we've seen before, and at least what I like to consistently point out, can't really select it, but you are not hydrolyzing, or at least you have your guanine exchange factor, as in when it goes from GDP to GTP, you are not phosphorylating. It's just like the GDP gets kicked out and your GTP binds. That is at least fairly somewhat critical to the pathway. And yes, you have, I assume they're going to elaborate on, okay, yeah. Well, they're pretty much going through your standard G protein coupled receptor process. Okay. So you have your receptor, your G protein receptor. You have your ligand binding. In this case, I'll zoom in more. Some sort of hormone, okay? And then, as they say, you have displacement of your GDP by the GTP, okay? And this results in essentially translocation, okay, to the adenyl cyclase. And that will catalyze a reaction that I assume they're going to get into in the next... Okay, yeah, I guess I'll stick here for now. I just want to make sure. Yes, GS is stimulatory G protein. They're also, yes... but yes, there are multiple subunits going back up here. Sorry if I'm scrolling too much. They don't really show it here. No. But yeah, the multiple subunits of, you have your gamma, your beta, your alpha, okay? And then, yes, once your GDP is kicked out and your GTP comes in, it is your alpha subunit, okay, that essentially translocates and interacts with your adenyl cyclase and will essentially turn it on or stimulate it. And let's see, yeah, 3 subunits, alpha, beta, gamma.
And whether it's a G, S, Q, or I, it's just the corresponding cascade of events.
Yes, it should. There might be some small twists on it, but yeah, this is this is the foundation.
Typically it still goes through cyclic or gets translated into cyclic AMP.
Yeah, I would say pretty much all of this is conserved. This is pretty much for every G protein coupled receptor, it starts with this cascade of events. And we're definitely, the GDP kicked out and your GTP comes in, and your alpha subunit translocating to the adenyl cyclase, which will, as the term implies, you'll make your typically your cyclic AMP. Yes, shown there. Yeah. So as the example of now they're using the epinephrine hormone as an example, which we've seen many times before. And yes, same thing. GDP comes in, GDP kicked out, and then now they're adding in that now you actually see it, that adenyl cyclase. Well, it cyclizes ATP into cyclic AMP, which we have seen before. Anything else? Diffuses away and binds. Epinephrine, yeah, they're just giving this as an example. Okay, and yes, here's where, there might be a little bit of a twist, but even then, I would say this is still, ultimately, what's occurring, 95% of the time. Stick with what's here, I would say. Don't deviate from this. But yes, cyclic AMP, okay, as they say, activates protein kinase A, and then that will result in a bunch of other phosphorylation events let's see. Okay, yeah, I'm going to go into more detail here. But yes, but cyclic AMP, as they're showing here, it can be degraded, and hopefully, no surprise, once it's degraded, then you are not activating protein kinase A.
Right.
And that's effectively it. Okay. keep going. I'm just trying to like I know I'm probably like speeding a little bit too much, but I'm trying to like just get through it and then like go back and do the review and all that stuff,
Right.
and then fill in all the details.
Mm-hmm.
Right now I'm just trying to cover as much ground as possible.
Yeah,
Okay,
makes sense.
so now they're using specifically your epinephrine receptor. Okay. Do phosphorylation by the beta adrenergic receptor kinase and subsequent binding of beta arrestin. Okay, so going through the sequence of events, no surprise, it all starts the same. Like every signaling cascade event is pretty much ligand binds to receptor, and your receptor is typically on the plasma membrane or the membrane of the cell. Although not always. Sometimes you just have you have your ligand, just entering into inside the cell, and like the receptor is not bound to the nucleus. But that's your niche one. But pretty much 90% of the time, your receptor is bound to the membrane, and you have your ligand bind to the receptor. And pretty much what they're showing here is like they're giving the example, like pretty much this is still a G protein coupled receptor. You have your beta subunit, you have your gamma subunit, okay? But now they're just like giving it a name of like in this particular pathway, this particular G protein coupled receptor pathway using epinephrine, okay? The BARC is playing the role of your alpha subunit or the beta ARC, okay? And then one moment, let me scroll more here. Yes, you essentially have your translocation, okay, or as they say, your beta arrestin binds to the phosphorylated carboxy terminal, one domain of the receptor, okay, so essentially a translocation event, and those phosphate groups act as essentially docking sites, okay, for your beta arrestin. Receptor arrestin complex enters the cell by endocytosis. So what they're showing here is pretty much, well, as it's described there. You get endocytosis, or the pinching in of the membrane, okay? Endocytic vessel, arrestin associates, returned, to this. So here you have this recycling event of pretty much how the receptor gets back or recycled back into the plasma membrane. Nothing overly complex here. Okay, neurotransmission. This is a good review. Again, as we've seen before, we have our membrane potential roughly at minus 60 millivolts. Okay, and that is maintained by this difference in charge across the membrane, as in pooling potassium and sodium, outside the membrane, and it's different concentrations versus inside the membrane or inside the cell. And that will result in your overall minus 60 millivolts membrane potential. And then they assume they're going to go through, your typical neurotransmission. Okay, yes, your acetylcholine receptors, which I assume you've seen from neuroscience, but, correct me if I'm wrong. I'm just scrolling forward just to see. Okay. Yeah, they go through the action potential and the passage of ions across the membrane and the influx and efflux. Okay. So, yes, acetylcholine reaches by motor neuron, diffused to plasma membrane by myocyte, binds the nicotinic acetylcholine receptor. And so, yes, there's in the receptor, just like we've seen for many receptors, okay, there is a conformational change associated with it, and that will result in the movement of ions, specifically positively charged ions, such as your sodium, such as your calcium, you probably already know this, again, from neuroscience and even from your own personal research of neurotransmission and muscle contraction, where calcium, I assume they're going to cover it, but maybe not. No, like calcium can also serve as a second messenger. Okay, I'm just trying to see how far that they... Okay, they go through systematically neurotransmission. Okay. So yes, it is ligand gated, so something needs to bind. Okay, and then yes, sodium flows down its concentration gradient. Okay, And then yes, you have pretty much you have the influx and efflux occurring, and your membrane gets depolarized, that you've seen before. But then yes, you also need to be repolarized as well. That I assume they're going to get into the nitty-gritty details of that. But yes, no surprise, once you allow the sodium and the potassium to flow, okay, your membrane potential changes. You go from minus 60 millivolts to plus 30. And then also, once the potassium flows out, you minus 75. So then to reset, to get back to minus 60 millivolts, you have to have that repolarization event.
Yep.
Which you've seen multiple times before. And yeah, this occurs very fast, as they say, less than 1 millisecond. Okay, so yeah, the propagation of an action potential. You've probably seen it before, where it's essentially like stepwise, or well, stepwise in less than a millisecond, right? But that's how we depict in the textbooks. Like we are propagating along the axon, and then like pretty much sodium moving in, potassium moving out, and then like what happens on one set of receptors stimulates it on the next set of receptors, essentially a domino effect to propagate across the axon, or to propagate the action potential across the axon. Yeah, so polarized, depolarized. Okay, yeah, so they're showing this is actually a pretty nice figure. In terms of...
Except for, and I think she did mention this going through the slides, but it... in hyperpolarization, obviously, like it needs the sodium potassium ATPase.
Yeah, pump. Yes, exactly. But yeah, they're thankfully also showing the change in membrane potential. So like you start at minus 60 millivolts, and like through the flow of the ions, your positively charged sodium and potassium, you become plus 30 millivolts, and then yes, at the end you get repolarized back to minus 60 millivolts. Yeah, you reset. The figure is consistent, as in you start with 3 sodium and 3 potassium in the beginning, and then thankfully, yeah, they're being consistent of at the end you end up with 3 sodium and 3 potassium, because now you are repolarized, ready to propagate another action potential. And like, yeah, there is that in your typical graph of an action tracking the membrane potential, you have that hyperpolar, that dip in the graph that you've probably seen before.
Yeah.
And yes, you have that inactivation part that's pretty important, where, as they're showing, with the sodium potassium channel, as you maybe know from neuroscience, like obviously there's a delay in terms of there's a limit to how much you can when an action potential is propagated, you can't like immediately afterwards, because there's that inactivation, as in like there's a little bit of a time delay, or that less than a millisecond time delay, but it is there. In terms of there's not an infinite capability to propagate an action potential. And like, yeah, it is that sort of essentially like that physical block, as in like, yeah, we are inactivated, we can no longer flow, we're physically at a molecular level blocking off the flow of ions, potassium and sodium. Okay, so then they go systematically, which is great. All right, your voltage gated both sodium channel, your voltage gated potassium channel, which, yeah, makes sense in terms of these respond to voltage and like well, the signals that that are received in your brain, among other places. In terms of, again, you have that threshold, as in you need to reach a certain threshold. It's all or nothing. It either occurs or it doesn't occur. There's no whatever spectrum. Okay, but once you reach that threshold, okay, so 1 second, I'm gonna go section by section. All right, so yes, you get depolarization pretty much now. It's just in the figure what we said in words, in terms of you have your flow of ions, okay, your sodium coming in, okay, and your potassium going out. Okay. And then, okay, yes, here's your neurotransmitters. They're using the example of acetylcholine in your vesicles. Okay.
Yeah.
And not, I guess, shown, but with your potentially your snare proteins, right? I assume they're going to go through sort of the release of the vesicles containing neurotransmitters, and then from the presynaptic to the postsynaptic, and then yeah, the release of neurotransmitters or the vesicles fusing to the postsynaptic cleft, and then releasing the neurotransmitters in the postsynaptic or the postsynapse. Okay, so let's see, anything else? Yeah. Oh yes. So yeah, this is actually somewhat important, or I don't know if your professor emphasizes it, but yes, this calcium, okay, that's recycled. If I remember correctly, okay, so the calcium is also released at the same time in in the synaptic cleft, right? And I believe one of the reasons for that is calcium plays a role in the snare proteins and synaptotagmin in terms of those proteins are dependent on calcium to pretty much like reel in and like mechanically fuse the vesicle into the membrane. I think
Right.
I assume they're going to.
So, but that
show
just gets released into the synaptic cleft and then taken into the presynaptic neuron, I assume.
Yes, that's what it's showing here in terms of you have your.
Okay, yep.
Yes,
I do see that.
exactly. But I believe the primary reason is that all the proteins involved for getting the vesicles to fuse to the postsynaptic cleft is, again, you have your snare proteins and your synaptotagmin of like those proteins. Again, they're essentially they are mechanically fusing the vesicle to the membrane, right? And
Right.
like
And was it
calcium.
calmodulin that activates that, or
No, calmodulin is a totally so calcium is involved in calmodulin.
Yeah.
But I do not believe
but that was
calmodulin.
through a different.
Yeah, that's a different pathway. But yeah, totally, there is an association between calmodulin and calcium. Snare regulated exotype, yeah, releases neurotransmitter into synaptic cleft. Yeah, exactly. Okay, now they don't really go much in the postsynaptic. Acetylcholine receptor ion channels, yes. And then, as you likely know, the neurotransmitters are also like recycled. Well, typically in a normal healthy human, they're recycled back. They don't just like stay there. They might be degraded, but they're typically like they're sent back to the presynaptic cell.
Yeah, either that or bound to acetylcholinesterase.
Yeah, that's which will degrade it because
Yeah.
because it's an esterase. Just to relate it to just like the thioesterase pretty much does that
Right.
cleavage. The acetylcholine esterase will also essentially break down the ester bond in acetylcholine. Okay, and yes, the actual receptors, what they look like. And yeah, no surprise, you have your three-dimensional receptors, but the ligand, in this case acetylcholine, bind at specific areas. I don't know how that's critical for you to know, but at the very least know that these are three-dimensional receptors. Just as you have an active site on an enzyme, you're going to have a ligand binding site on your receptors.
Right.
Nothing super special there. Yeah, 5 subunits with 4 alpha helices each. Amphipathic helices around. Yeah, okay, there's nothing, say, super critical there. Okay. They're going through the opening and the closing. Interesting. Oh, that's cool. So, well, no surprise, or potentially no surprise, is so yes. Since this is a protein and it has a three-dimensional structure, okay, its structure is, it's in whatever, intentional, or it's not, an accident, as in it adopts a very specific three-dimensional orientation. And so just as we saw with other receptors in the membrane of, like, generally speaking, if you're a protein embedded in the membrane, okay, then your hydrophobic amino acids that are exposed to the membrane, okay, they're going to be exposed to the membrane. And then your hydrophilic amino acids that are more on the outside, they will be like on the outside. And then if you're looking at, let's say, a channel protein, like the aquaporin, well, what allows the aquaporin to be specific to water and a water molecule is the three-dimensional conformation that the amino acid side chains, okay, play a role in coordinating the water through the aquaporin. Same thing here, same principle here, but what they're showing is pretty much how the leucine side chains, okay, play a role in terms of, like, these leucine residues are critically important to the overall functioning of the acetylcholine receptor. And let's see, yeah, these and leucine hydrophobic, where yes, they essentially, as they're showing here, they close the channel and they're essentially moving, as in once the ligand acetylcholine binds, they essentially move out of the way, where you have this conformational change as a result of acetylcholine binding.
And it does bind to acetylcholine molecules per receptor.
Yes, exactly. Where here you have so it's 5 alpha helices.
And then the 2 alphas helices, are where it binds.
And 5 subunits. Yes, exactly. I mean, I don't know if it's super important to know where they bind, as in you need to know that they bind between between the subunits.
Yeah.
Definitely, you almost certainly need to know that there are 2 acetylcholines that bind, and that there is a closed and open conformation, which hopefully is intuitive in terms of, well, this is an acetylcholine receptor, and so hopefully it makes intuitive sense that it will change into the open conformation upon the binding of acetylcholine. In this case, 2 acetylcholines.
Yeah.
Okay, and what they're showing here is how not only, at least based off of this figure, it's like in the closed conformation, like well, one, you have your leucine side chains that that looks like it's creating a physical block, but also the fact that these are hydrophobic, it has a specific chemical environment in the interior. And then what they're showing here at the end of the open conformation, when those 2 acetylcholine ligands bind, okay, then actually the chemical environment within the receptor, let's say within the channel or the pore of the receptor, actually changes. As in you're going from hydrophobic on the left to polar or hydrophilic on the right, which it's almost certainly the case that promotes the passage of the ligand through the acetylcholine receptor, potentially. Okay, receptor enzymes, which again we've seen before, but all we're really going to essentially see, as we've seen before, is a receptor that will carry out some sort of chemical reaction, an enzymatic reaction. Which the tyrosine kinase, we've seen the guanylyl cyclase, we've seen the tyrosine receptor kinases before, of those autophosphorylation events on the tyrosine residues. But there are other varieties of receptor enzymes. Okay, so they're using the insulin receptor, thankfully, as the example. Okay, so one, you have your insulin, small peptide. Or fairly small peptide relative to a full receptor. But yes, you have insulin binding to its respective receptor, which in this case should be the insulin receptor specifically. Okay, and again, if you maybe recall, although I don't necessarily expect you to, for the tyrosine receptor kinases, you kind of get this dimerization of, as a result of the ligand, in this case insulin binding, you're sort of dimerizing or bringing the receptor closer together.
Yeah.
And then yes, you get the autophosphorylation events. Like they're essentially phosphor. So I'm pretty sure. So here they're showing the phosphorylation. I'm pretty sure that when they say autophosphorylation, it's like it's happening like across. I don't think it's happening as in like where those phosphates are added. It should not be from the one above. You can see, hopefully you can see that you have two that look identical. I'm pretty
Yep.
sure it's sort of like a cross phosphorylation.
Well, I think that cell biology course slide deck had a
yeah,
bit better of a figure show that.
yeah. I'm gonna try and bring it up because even I myself, I was like, because when they say autophosphorylation, I think it's like whatever. If we're being really pedantic, it's strictly speaking not autophosphorylation, but
Yeah.
gonna bring it up. One moment.
I want to say it was signal transduction 2.
Well, I have that. Yeah, I definitely remember these slides. These are pretty good slides.
Yeah.
Uh,
3.
yeah, I have 3 as well.
Yeah, I think 3. Although I think he was using EGF instead of insulin, but it's similar. Oh, and then
But yeah, but just to not answer, but like as you brought up, so you're right. Like as mentioned, like calcium is associated with calmodulin, as shown here, but it's not strictly speaking part of that pathway they were just covering. Yeah, okay, tyrosine, EGF, yeah, same thing. Okay, yeah. That's why I'm a little bit iffy on when they say autophosphorylation.
Yeah.
Because like, so these are so let's say starting here. Like these are like they dimerize, right? As in they're separated, and then like when you have ligand binding, they come together now. And then
and a dimer is just like together. Like, is that basically the definition, or
strictly speaking, a dimer is two separate. When I'm talking about the protein world, a dimer is two separate polypeptides coming together through non-covalent interactions. That's what it would strictly be. As in, in each respective one, okay, like each whatever strand. That's one single polypeptide. At least it should be one single polypeptide, as in from beginning to end, amino acids linked together, and then you have your tyrosine, in this case 5, like linked together. So that's all through covalent bonding, okay. And then that's with dimer, trimer, or tetramer. So I'd say your canonical example of like think of hemoglobin. As in hemoglobin has your alpha and beta subunits, okay?
Right.
And that has
tetramer of 2 alpha and 2 beta.
Perfect. Yes, exactly. But it's 4 separate polypeptides like coming together to assemble. So that's a tetramer, okay? A dimer would be like just two polypeptides, okay? And yeah, when hemoglobin comes together into that tetramer, it is through non-covalent interactions, specifically like hydrogen bonding, and all your other non-covalent interactions that even for whatever reason is like escaping my brain. London dispersion forces, hydrophobic interactions, all that stuff. So when we're saying here dimerize, it's like they start interacting, and they start interacting through non-covalent interactions. And then yes, you have your autophosphorylation. Now, is it okay? Let's bring up 1. Like, is it when they dimerize? It's like this one goes back and phosphorylates all those 5 tyrosine residues there, or is it that it does the one on the opposing one, where I can consider that an autophosphorylation event because we've dimerized now, right? And so
Right.
just as hemoglobin when it's a tetramer, it's like one single entity, but
Exactly.
it's still a tetramer. That's why I'm sort of this autophosphorylation thing always gives me the runaround in my brain,
Yeah.
where I'm pretty sure it's like opposing, as in one phosphorylates the other, but since they're in a dimer, we call it an autophosphorylation event. I'm reasonably sure, although I'm pretty sure that detail's not even all that critical for you, but you definitely do need to know that it's an autophosphorylation event for sure.
Yeah.
Okay, and yes, opens up the active site. I assume they're gonna go more systematically. Okay, and yes, again, and it's not a surprise that it's on the tyrosines because the tyrosines have that OH group.
Yep.
Oh, 1 second. Okay, where yeah, it's not a surprise that it's on the tyrosine because it has that OH group. Okay, so when the insulin receptor is inactive, the tyrosine is kind of blocked by its activation loop. Okay, so common theme where so it's a kinase, right? And so it phosphorylates, and whatever we say that it autophosphorylates. And so if it autophosphorylates and it carries out, it has catalytic activity, specifically a kinase, so it phosphorylates. Well, we don't always want to phosphorylate, and this is like it basically it self-regulates, okay?
Right.
Where you have pretty much we're going to essentially sort of like be blocking off the active site, okay? Where yes, this loop has 2 tyrosines, including one which makes a key hydrogen bond, yeah, with an aspartate, okay? Where yeah, well, essentially what they're saying here is that interaction between the tyrosine 1162 and the aspartate 1132. Okay, that interaction when it's present, like essentially like blocks off catalytic activity, or basically you're in your inactive state. You can't carry out
Right.
your kinase activity.
And it's only when they're brought together that it
Yes.
basically changes the conformation
Yeah,
and
exactly.
allows it to phosphorylate itself.
Exactly. And like that's another example. Yeah, hydrogen bond. That would be a non-covalent interaction. And yeah, that is also hopefully recognizing the common theme of like when a ligand bind, just as when a substrate binds with an enzyme, there is typically or very often some sort of conformational change, built in such that, now it activates it. Just as like when insulin binds to the receptor, in this case now it's a tyrosine kinase receptor, that will induce some sort of it'll induce dimerization. It would also induce, well, as when they're dimerizing, there is almost certainly conformational changes associated with that. And that will almost certainly result in the breaking of that interaction, that hydrogen bond, that will then, turn it on to into its active form, and then start autophosphorylating. Those would be the sequence of events in a nutshell. Okay. So yeah, all 3 tyrosines, let's see, triply phosphorylated activation loop moves dramatically, making room for the target protein, yeah, in the substrate. But yeah, so exactly what I just said earlier, like there's going to be some sort of conformational change, and we're pretty much like before in the previous slide, it was blocked. The active site was blocked by that interaction between the tyrosine and the aspartate, that hydrogen bond. And then yeah, once there's those conformational changes, it's like that bond is broken, and now there's room for the substrate to bind. Substrate proteins can now bind and be phosphorylated, or we can now autophosphorylate each other.
Right.
Okay. And yes, no surprise, going down the signaling cascade, where we are up here. Okay. And So yeah, we are going to eventually end up in the nucleus, and we'll be altering a gene expression. And like, yeah, you have, I'm pretty sure you've seen these before. These confusing things, the
Yeah.
MEK, the ERK. Thankfully, I don't think you're covering like MAP kinase and MAP kinase kinase and MAP kinase
No, I
kinase.
don't think so. I mean, honestly, it's not like too difficult once you kind of see it's like,
Yeah,
complicated but simple at the same time. Like they named it to make it easy, I guess, at least.
yeah, for that one, like for sure. Like yeah, you're right. There's less issue with that one. Like, but I definitely don't like ones like that, or they're either MEK or ERK. And then
Oh yeah,
I hate like RAF and RAS and all that stuff.
easy to flip them around.
Yes, exactly. But going through it, systematically. Okay, same thing. You have insulin binding to the insulin receptor. In this case, the receptor is a tyrosine kinase receptor. You get your dimerization. They're showing that nice dimer forming, and then yes, you get your autophosphorylation events. Okay, and then this will result in the phosphorylation of IRS1. Okay, you pretty much get your cascade of events, or your domino effect of IRS1, GRB2, SOS, and RAS and RAF. But here you have, this should also be a guanine exchange occurring, as in GDP kicked out and
Right.
GTP coming in.
And is that the same as like a G protein coupled receptor, like it's GDP plus an inorganic phosphate, or just
Yeah,
GDP?
like the GDP that you find here is going to be the same GDP that you find in the G protein couple. Yeah, it's going to be
Okay.
guanosine. Guanosine diphosphate or guanosine triphosphate. Yeah, exactly. And well, yeah, you essentially get a bunch of phosphorylation events. Of MEK becomes phosphorylated, diphosphorylated, and then ERK becomes diphosphorylated, and ERK looks like, yeah, it's going to activate some transcription factors. Okay, as in your transcription factors are going to be your SRF and your ELK1, and then that will alter gene expression, either upregulate, well, typically upregulate, as in turning on transcription, but it's not always the case. It's stimulate transcription, translation of a set of genes needed for cell division, or whatever, some sort of outcome. Let's see, anything else here? RAF1, yes. Well, yeah, no surprise, again, just to reinforce the concept of, yeah, you have RAF1 phosphorylates MEK onto serine residues, because again, serine has those two OH groups that are perfect sites for phosphorylation. Okay. And yes, no surprise. So like, we make the association, understandably, and even probably most people like on the street now these days know that insulin plays a role in regulating blood sugar. And like, yeah, when insulin binds its insulin receptor, okay, yeah, you also have the pathway of like that will result in glucose transporters being transported to the membrane that allow glucose to come in. But then, as they're showing here, okay, the insulin receptor, we're not just regulating the gene expression for glucose transporters, right?
Right.
As in, you have all these other things going on, where, well, these are the cascade of events, but these transcription factors are probably not only just like turning on the genes for the glucose transporters. Okay, that's probably like one of the things. But even as they say here, we're starting with insulin, right, all the way at the top. And then, yeah, we're resulting in transcription factors, in the cell. And then, yeah, at 0.7 there, they say translation of a set of genes needed for cell division, in terms of like what's the connection between insulin and cell division, right? As in, all that to say is like, there is a role there for insulin and cell division, presumably, at least based off of what they're saying there. and not just, upregulation of the genes required for glucose transport or getting glucose inside the cell.
Right.
So just reinforcing the point that, just as one ligand very often is not responsible for like one type of signal, one receptor is typically not responsible for just one type of signal. There's likely like multiple different outcomes. Okay, yes, glycogen synthase, and yeah, glucose transporters by insulin. So now they're getting into, and well, yeah, we've seen glycogen synthase or glycogen synthase kinase 3. And its role in making glycogen, which we're totally going to revisit. So, going through the sequence of events. So yes, IRS1 phosphorylated by the insulin receptor. Okay, pretty much. Let me, I guess, zoom out a little bit more and get them all three on the screen. Okay. So, you have a bunch of things occurring, again, through all these signaling cascades, okay? So, let me just double check here. 3 on serum residue. So, the ultimate endpoint, okay, so to speak, is, yeah, in this case, we want to get glucose transporters to the cell. Okay, and GSK3 plays a role in glycogen synthase. Okay, so what they're showing here is glycogen synthase. When it's phosphorylated, it is inactive. Okay, and then when glycogen synthase kinase 3. Sorry, I need to be very specific. GSK3 versus GS. Right. Where GS is there on the left, that's the glycogen synthase. That will be a key player in the synthesis of glycogen. So make sure I get my terms very clear. Glycogen synthase kinase 3, when it's phosphorylated, it is inactive, okay? And then when it's dephosphorylated by a phosphatase, well, that's going to play a role in essentially glycogen synthase, of basically turning it either on or off, as in glycogen synthase, okay? It's essentially like a circuit. When glycogen synthase kinase 3 is phosphorylated, okay, it is inactive. When it gets dephosphorylated, okay, that means now it has its kinase activity, okay? So since glycogen synthase kinase 3 has its kinase activity, then it can phosphorylate glycogen synthase and deactivate that. As in, basically, the active GSK3 turns off or inactivates glycogen synthase. Okay, right. And where, yeah, it's, at least for me, difficult to like wrap the logic around it in my mind. But at least for both of them, they are both inactive when they are phosphorylated. As in GSK3, phosphorylated inactive. GS, phosphorylated when it's phosphorylated, it's inactive. And like, yeah, they both become active when they are dephosphorylated. But then the GSK3 is phosphorylating the glycogen synthase and effectively turning it off. Yeah, very complicated and confusing.
Yeah.
But then, but definitely, I guess, the way that I try to anchor myself doesn't always work. Of like, glycogen synthase kinase 3, it's a kinase, so it's going to phosphorylate. As in, if you know, as long as you, I guess, keep in mind that there's a relationship between glycogen synthase and glycogen synthase kinase 3, and that at least you know for this, that the being phosphorylated, that means you are inactive, okay? So then, when GSK3 is dephosphorylated, it's now active, okay? And now that it's active and then GSK3 is active, it's a kinase, well, now it can carry out its kinase activity. And so then it can phosphorylate glycogen synthase, and that inactivates glycogen synthase. That's how I sort of attempt to anchor myself. But how do we get there to begin with? Okay, so backtracking. At least I typically like to backtrack with pathways. Okay, how is it that GSK3 gets dephosphorylated? Well, then the protein kinase B, when it's phosphorylated, will essentially acting as a phosphatase, removing that phosphate group from GSK3. Okay. And then yeah. No, sorry. I'm not following the direction of the arrows. Sorry. Yeah. The direction of the arrows with PKB and GSK3 is yeah. Since the arrows are going back from the active to inactive, and PKB is playing a role there, it's the protein kinase B that is phosphorylating GSK3 and maintaining it in its inactive state. Okay. And yeah, PKB stimulates movement of glucose, where ultimately our endpoint is getting glucose transporters to the membrane. And then yes, when glycogen synthase is active, as in it's dephosphorylated, then we're going to be making glycogen, which we are obviously going to revisit. Okay. Insulin acts in muscle cells to, I assume, well, yeah, this is your typical, I guess, one of your symbols for active or upregulate.
Yeah.
Okay. Active glycogen synthase by phosphorylation of GS. Okay, pretty much everything that we just went through. Of GSK3 phosphorylates GS, and then when GS is phosphorylated, that means it's inactive. But just as this is sort of a refresher of glucose, and how does glucose get incorporated into glycogen? Okay, well, the main form of glucose into glycogen is UDP glucose, as in that's basically our building block, or what we're going to need to use to make our glycogen. Okay, our branched polysaccharide of glycogen.
Right.
And all of this stems from glucose to glucose 6-phosphate from hexokinase, which we have seen in glycolysis. But then that in and of itself needs to be rearranged. This is where the mutase should come into play. We're going to move the phosphate group from position 6 on glucose to position 1 on glucose, and then that gets activated into UDP glucose, which will then be incorporated into our glycogen polysaccharide. But this is just, glycogen synthesis in a nutshell.
Yeah.
Okay. Yes, so what I just briefly went through when it comes to like dimerization, okay, as in that's dimerization when the tyrosine receptor dimerizes. Okay, that's like effectively sort of that's a polypeptide interaction, but to form the full dimer protein. But that's all mediated by non-covalent interactions. And like, yeah, protein-protein interactions are essentially all mediated by non-covalent interactions as well. And activation, or yes, well, we've seen that before, and that's something that I try to emphasize. Of phosphorylation can either activate or inactivate. This is just summarizing everything that we just went through. Yeah, reduced phosphorylation of glycogen synthase, increased activity, as in you are active as glycogen synthase when you are dephosphorylated, and then backtracking. Well, then the GSK3, the kinase, if it's actually carrying out its activity, then it's phosphorylating glycogen synthase and then turning it off.
Right.
Movement of glucose, yes. Okay, this is just a summary of the slides in words. Okay, nuclear hormone receptors. Yeah, so this should be, I guess, sort of intracellular signaling, where now you're going to have your ligand, like, should be entering the nucleus, and then you have a nuclear receptor that's going to, once the ligand binds the receptor in the nucleus, you're going to get your cascade of events. But ultimately, okay, pretty much transcription factors, or pretty much doing something to transcription factors that will alter gene expression. Very often turn on, but also potentially downregulate. Okay, yeah, steroid hormones, thyroid hormones, all these things that we've seen before. Okay. Yes, so this is thankfully a little bit more simple. Okay. You pretty much have your hormone binding its respective protein, in circulation potentially. But basically, yeah, some sort of serum binding protein with your hormone that will pretty much diffuse across the membrane. And going back here, so steroid hormones, and they're using steroid hormones as an example of nuclear hormone receptors, as in they are essentially crossing the membrane without a channel, right? As in,
right, straight into the nucleus.
Yeah, exactly. Where if you follow the arrow, it's like piercing the membrane, right?
Yeah.
And that let's say that makes biochemical sense in terms of, well, these are steroid hormones, okay, derivatives of cholesterol, and so if they're steroid hormones, then they are hydrophobic, okay, or essentially largely hydrophobic, and if they're hydrophobic, then they can potentially like pass through the membrane like on their own, and they don't need like potentially a dedicated transporter to get them through, like something that is charged or polar. And then same thing here. Although here they're actually showing it of like there's a dedicated or at least there's some sort of pore in the nuclear membrane that allows it through, or it's just a coincidence that the arrow is happening there. But at the very least, pretty much for sure, at the plasma membrane of the cell, like it's not absolutely necessary to have a channel to allow the hormone to come in, because it's hydrophobic, and therefore it has some ability to pass through the membrane or diffuse through the membrane on its own. But once it gets inside the nucleus, well, then there's a receptor potentially there waiting for it, okay? And then, well, yeah, you'll get binding, or rather, the nuclear receptor in complex with the hormone, then that will result in pretty much, the alteration of transcription. Where here it's a little bit more, I would say, elegant and simplified, of just hormone, maybe from circulation, passing through the plasma membrane, getting to the nucleus, binding its respective receptor inside the nucleus, and then changing.
And it binds to heat shock proteins, doesn't it?
Some of them will. It should not be universal, as in every hormone. But that is a potential outcome. Do they mention heat shock proteins here specifically? Just want to double.
I don't think so. They said receptor proteins broadly.
Yeah.
In under one, but nothing to do with heat shock proteins.
Yeah. But that is a
potential. I went over that like briefly in physiology.
But yeah, I mean, heat shock proteins like are a thing, like for sure. But I don't think they should not be. Well, yeah, not every hormone binds a heat shock protein. That is for certain.
Yeah.
Okay, and the structure of the overall structure of a nuclear hormone receptor, and this is actually fairly important. So, going back here, it's maybe not super obvious, but a little bit zoomed in too much. When your hormone okay, binds to its respective nuclear receptor, okay, you get essentially a complex, okay, and that complex, as they're showing here, where's my, there, let me scroll, as they're showing here, like, so you have, well, multiple things. You have your DNA, you have your receptor, and you have your hormone binding to the receptor, so you have like a whole complex here, okay. So then you need specific regions on the receptor devoted to specific things and functions. As in, if this is going to act as a transcription factor and like do something to the DNA, then it needs a DNA binding domain, and that will be separate. That will be separate from like the binding domain for the hormone. Okay.
Right.
And they're going to, looks like they're going to describe of like, well, what does a DNA binding domain on a nuclear receptor look like, or what are some examples of that? Okay, so yes, these zinc fingers. Okay, so that's essentially the part that allows this this nuclear receptor, when it's bound to its respective hormone, that's what allows it to bind DNA, and thus alter transcription, or play a role in altering transcription. As in these zinc fingers, which are these motifs. And they call them zinc fingers because, yes, they do have zinc metal in them. They are coordinated by these cysteine residues, okay? So ultimately what would be happening at a broad level is your hormone, passing through the plasma membrane, your hormone getting into the nucleus, okay, and then your hormone binding to its respective hormone receptor in the nucleus. That will almost certainly, more often than not, result in a conformational change, okay, where now these zinc fingers can go and do their thing, or they now have the ability to bind to DNA, as opposed to without, in the absence of the hormone, the zinc fingers are likely, or rather, the receptors in likely a conformation that does not allow these zinc fingers to bind DNA. So again, a lot of it comes down to conformational changes. And yes, they are zinc fingers because they have zinc, and like they are actually binding DNA. Okay. Yeah. So they are showing here in terms of the overall structure of the polypeptide or protein.
So these would depend on, what's binding, I assume.
Yes. Yeah. When you say the zinc fingers.
Yes.
Yeah. Yes. As in, it will depend. As in, like their orientation, as in they're going to have to adopt, and there's a reason why they call them like zinc fingers, because they sort of like resemble like fingers, at least in their active form. Where basically like in the absence of the hormone, your zinc fingers will not be in the conformation, a stable conformation to bind DNA and do its thing. And that's why you need separate domains. What they're showing here, that is color coded, like on the right, you have your hormone binding domain, as in like this is the region on our protein, or on our receptor, which is a protein, that's responsible for pretty much interacting with the hormone. Okay.
Right.
And then when it binds there, in all likelihood, that will result in some sort of
being red in that section.
Well, yeah, that's one way to put it. In terms of it will likely alter the 3D orientation of the zinc fingers, as in they're there, as in like this sequence of amino acid is there, okay, but it's likely not in the correct orientation in the absence of the hormone.
Right.
But then in the presence of the hormone, just as I guess maybe think of hemoglobin, of like when oxygen binds hemoglobin, and when oxygen binds hemoglobin, it's cooperative, right? As in when a little bit of oxygen binds, that allows it to bind even more oxygen, and you have that sigmoidal binding curve of it because it's cooperative. Where when a little bit of oxygen binds, okay, the structure of hemoglobin shifts a little bit. That allows for more oxygen to bind, and it's sort of like well, it's cooperative in that it's helping itself, so to speak. Or as oxygen binds, it becomes in a more favorable conformation to allow more oxygen to bind. And so analogously, when in the absence of the hormone, just as in the absence of oxygen, hemoglobin can still bind oxygen, but it cannot bind oxygen as well as when it's like 50% saturated, because
Right.
there's a conformational change. And so when the hormone binds, there is almost certainly a conformational change, where you can kind of think of it as like, well, in the absence of the hormone, the zinc fingers are not exposed. They're like in a fist, so to speak. But then
Okay.
when the hormone binds, okay, there's conformational change that now you're no longer in fist. You're actually like whatever, your fingers are spread out. And those can go and bind DNA and act as, well, a transcription factor. That is ultimately would be happening. And yes, and you have a transcription activation region on the left there, where yeah, what they're doing here, what they're showing here in the color coded is like, this is the entire sequence of the polypeptide from amino terminal end to carboxy terminal end on the right. If I scroll there, and they're just they're just showing how there's like different sections of like, here's the section of the proteins, amino acid like 1 to 20. This is it serves this function, and then amino acids.
Oh, okay. You know what? Straight up, I had totally thought that that was illustrating a DNA strand at first.
Oh, fair. Yeah, fair enough. No, I
Yeah.
understand. I understand.
So that makes much more sense now.
Okay, good. I'm glad.
Yeah.
Yeah, these things. That's why I understand why they depict things as like this. Just like I know why they depict proteins as misshapen blobs.
Yeah.
But then it becomes understandably difficult to, like, I guess, fully understand or know, like, well, what are we talking about? Like, again, if this isn't labeled, how do I know what it is, right? Unless
Yeah.
I'm in the habit of like, oh, this misshapen blob, that typically represents a protein. And this protein seems to have something tethered to it or scanning through it, and so this is probably like mRNA, but totally in a vacuum. Like looking at this, it's not immediately obvious, and that's why they're trying to at least sort of, I would say, poorly signal to you that there's an NH3 there on the left and a COO minus, so this is an amino acid sequence.
Right.
And that yes, there are sections to this protein, and specifically, thankfully they number it, like the amino acids 66 to 68. Those residues are highly conserved, and they're basically showing you
Right.
here the amino acid sequence in that region, presumably. This looks like a full amino acid sequence. I mean, this would be like methionine, lysine, aspartate, tyrosine, or blah blah blah.
Yeah.
Okay, great. Oh, well, that was the last slide for this lecture. Okay, I
Perfect.
was ready to scroll more. Okay, so then yeah, let's start reviewing, getting to the nitty gritty details, and start refining things and hopefully solidifying things in your mind. Okay,
Yes.
so I think probably
And
maybe.
I'm just gonna run to the
Yeah,
bathroom
yeah,
real
go for
quick.
it. Yeah, of course. Yeah, anytime, for sure.
Right.
All right, so going back to gluconeogenesis, fatty acid stuff. 1 second. Well, so you're not tested, I think, on glycolysis specifically, as in like you don't need to memorize those steps.
No.
Right.
No.
But like you definitely do need
Only
to know.
yeah, like the major steps 1, 3, and 10.
Yeah, great. And I think you probably need to know in a little bit more detail of gluconeogenesis, at least based off the impression that I get. Okay, so well, starting with I would say so obviously we'll go through it in more detail, you know, and I'm gonna probably like start quizzing you more and more, but.
Yep.
If you want to just, you know, as best you can off the top of your head, it's like, well, okay, glycolysis and gluconeogenesis. Like, what are the? I don't need like obviously the 10 steps of glycolysis from beginning to end, but let's say, you know, what's the overlap between glycolysis and gluconeogenesis, and like, you know, what's unique to gluconeogenesis if you can, and like, well, again, you don't have to do it off the top of your head, but just to like you know, get in the habit of like repeating it and sort of solidifying it in your mind.
Well, I know that like, I mean, it's hexokinase / glucokinase, phosphofructokinase, and pyruvate kinase, which are the main why does that word always escape me?
That's okay. No, it'll come. And then you can use your, like, it doesn't bother me, because like
I'm very much using my notes, don't worry. But no, I it's the but you know, you can't go back on the steps.
The irreversible steps.
Irreversible. There you go.
Or if it's easier, like I actually prefer like committed steps.
Yeah, committed steps is a better way to say it.
But same thing, like whatever you prefer. If it's easier for you to remember committed steps, then like, you know, go with that, whatever's easiest. And like, yeah, so are you able to like tease out or just like say, what are the committed steps of gluconeogenesis?
Which are just basically reversing those committed steps. The 2-step process in the pyruvate kinase, which is reversing phosphoenol pyruvate into I think it's basically 2-phosphoglycerate, but it's going through pyruvate carboxylase and phosphoenol pyruvate carboxykinase.
Yes.
Which are using ATP as opposed to you know, generating ATP.
Yeah, that is critical in terms. So yeah, for glycolysis. So you know, in your steps, you have in your first phase, you have the consumption of ATP, and then like, yeah, you have your payoff phase in like the second half, or you know, the last whatever 4 steps. And like, yeah, but in the reverse, in gluconeogenesis, you are consuming ATP. Yeah, keep going. Yeah. Great.
And then specifically, I know that in that first step of gluconeogenesis, going from pyruvate, you have to go into oxaloacetate first, basically in the mitochondria, preserving NADH, I believe, as well.
Yes, that sounds absolutely right.
Yeah, and I believe in going to oxaloacetate, yes, that's the one step where CO2 is consumed.
So ultimately, you are right. I just want to actually clarify sort of like one thing in terms of like CO2 is involved there. It's not strictly speaking like CO2 the gas, but like whatever. But CO2 is fine. I don't want to like overcomplicate things, but like, yeah, you're right.
Okay.
That CO2 is leaving. I was just trying to make the distinction that the enzyme is not dealing with a gas. It's actually in the form of hydrocarbonate, HCO3. But it's like through the
Right.
use of that, CO2 is being removed. Or sorry,
Okay.
added in as in to make oxaloacetate, pyruvate to oxaloacetate.
Right.
Yes, you are carboxylating. But it's not strictly speaking, it's getting to the weeds of like it's not using CO2 as a gas per se.
Right. Okay. Yeah. And then
Yeah, keep going. Great.
going from oxaloacetate into phosphoenol pyruvate, use phosphoenol pyruvate carboxykinase, which is essentially what it's doing. I believe that's where CO2 exits again.
Yeah, so that is true. Yes, exactly right. And there's another thing that's involved there that is pretty important, as in like you're right on the money in terms of phosphoenol pyruvate carboxykinase, and CO2 is produced. Yep. But there's another sort of pretty important thing that occurs in terms of like this step requires something.
Requires GTP.
Yes, exactly. Perfect.
Yeah.
And so yeah, that's at least in terms of the differences between glycolysis and gluconeogenesis. Like that's the beginning. It's different. Everything else proceeds.
Until step 8 of gluconeogenesis, which is at fructose 1,6 biphosphate, which is converted through fructose 1,6 biphosphatase 1.
Yes, exactly. Very, very convenient.
Yep. And then again, proceeds like normal to glucose 6-phosphate, which is converted through glucose 6-phosphatase into glucose.
Can you repeat that one more time? Sorry.
So fructose 6-phosphate proceeds as normal to glucose 6-phosphate, which is goes through glucose 6-phosphatase into glucose.
Okay, yeah, perfect. No, that's great. That's exactly the sequence of events. But then I don't know if it's important for you to know, but like maybe keep in mind like that fructose 6-phosphate to glucose 6-phosphate. There's an enzyme there that I don't know if you have to know, but just to I guess cover all the bases of like there's an enzyme there. There's an isomerase.
Isomerase.
As in, so the fructose 6-phosphate to glucose 6-phosphate, it should be a phosphoglucose isomerase that carries out that reaction.
Okay. Fructose 1,6 biphosphatase.
You're right in that fructose 1,6 bisphosphate is converted into fructose 6-phosphate, and that is by fructose 1,6 bisphosphatase. But then
phosphatase.
Yeah, and so right now, like we're at fructose 6-phosphate, and then that goes to glucose 6-phosphate, right?
Right.
But to carry out that reaction, because like glucose and fructose are different things, right? Even though they're both 6 carbons, and even though like the phosphate is still on the 6th position, as in we're going from fructose 6-phosphate to glucose 6-phosphate, but there is an enzyme that has to carry out that reaction, as in it's basically it is an isomerase. It should be phosphoglucose isomerase that carries out that reaction. Whether you need to know that, I don't know, but I'm just trying to make sure that I'm covering all the bases.
Okay. And is it also known as phosphohexose isomerase?
It could be. It would make sense. Because a hexose is just a 6-carbon sugar. Okay, you might then, if that's what's in your notes and textbook, then like go with that. I just know it as a phosphoglucose isomerase.
I mean, I think phosphoglucose isomerase is easier to remember because it's going to glucose 6-phosphate.
Yes. absolutely.
I know in the slides that's what it says.
Uh,
I
okay.
think is pulled from probably Leninger 6th edition.
Yeah, I think most of the stuff here is pulled from literature. Okay, great. So yeah, pretty much you know the differences between glycolysis and gluconeogenesis, and just pretty much the differences in those three steps. Two at the
Yeah.
end, one closer to the beginning. But then, like, yes, obviously, regulation is important. Okay, now, do you have in mind the overall themes of regulation in terms of like what overall regulates either glycolysis or gluconeogenesis, and like they're reciprocally regulated, right? As in like what stimulates one or activates one is likely going to inhibit the other. Obviously, I'm not looking for all the regulators, but like, are there any major regulators that you're aware of in terms of, well, this activates gluconeogenesis and will inhibit glycolysis, or vice versa, or like what are the regulators that you know of?
Right. So definitely glucose
Yeah.
would put it in favor of glycolysis, as well as I think fructose 6-phosphate and um, fructose 1,6.
There's
Uh,
one of the
Bisphosphate.
bisphosphates. Yes, it's
Yeah.
even I myself. It's either 1,6 or 2,6. Which, yeah, we
Oh
are
yes.
going.
It's 1,6 and that gets converted into 2,6 by an enzyme, and 2,6 is the like major regulator of phosphofructokinase.
Yes, yeah. Oh, great, perfect, wonderful. And so yes, hexokinase. Since you have a lot of slides devoted to hexokinase, so like that is regulated in a key way, and that makes sense since it's one of the first steps in, well, it is the first steps in glycolysis of glucose to glucose 6-phosphate. Now, do you remember if you're able to say like, well, do you know what we mean when we say allosteric regulation, and like how it's allosterically regulated?
Uh, yes, it basically binds to it, giving it higher affinity.
Yes, yeah. Well, so when you say what specifically binds to it, and just to
So, like, say like glucose or fructose 2,6 biphosphatase would bind to um, phosphofructokinase to like
uh,
regulate it positively, I believe.
yeah, well, if you're saying activated, then yes, it would be positive regulation.
Yeah.
If you refer to inhibition, then like, yeah, it would be negative regulation. But for hexokinase specifically, like, do you happen to recall in terms of like how it's allosteric? Well, it's even here, where just to reiterate, the concept. Do you know specifically what we mean when we say allosterically regulated?
So I believe glucose for say hexokinase is a positive homotropic allosteric regulator,
Mm-hmm.
uh, where it binds to it, increasing its activity.
Mm-hmm. Well, so, and just to, I guess, make are you saying it binds like in the active site, or like what's the concept of allosteric regulation? Just so you're familiar, like, with that concept.
Allosteric could bind to like whether it's heterotropic or homotropic, would bind to either the site itself as the substrate, allosterically regulating it, or an allosteric site uh, outside of the actual substrate binding site.
Yeah. So, perfect. Wonderful. Although I think generally speaking, like when we allosteric regulators would be, I think most of them should be different sites than the active site. But like, I'm glad that you're able to like make that distinction. Like, that's perfect. Um,
Well, the
Okay.
only reason I say that is because I literally just looked up homotropic and heterotropic allosteric regulation definitions.
Oh, okay. Well, great.
Yeah.
That's a high level of detail, which is absolutely a great thing. Okay. So yes, as they're showing here, they're showing two different versions of hexokinase. Okay,
Yeah.
and they're talking about the K0.5. Okay, so do you know what that means in terms of, well, are you able to explain sort of the concept of K0.5? Although I don't necessarily expect you to remember, but as much as you can.
Somewhat. I know the lower K0.5, the higher affinity it has for the molecule. So it needs
Yes.
a lot less to basically reach its max velocity or
Yeah.
activity.
Yeah. Pretty much. You can say maximal velocity or maximal enzymatic activity. Yeah, both like
Yeah.
work perfectly. And yeah, perfect. That's the concept in terms of, so yes, you have hexokinase 1 and you have hexokinase 4.
Yeah.
And as they're showing here, they will have a different K0.5 in terms of like the hexokinase 1. You need a much lower glucose concentration to stimulate its enzymatic activity, as opposed to
Right.
hexokinase 4. Okay, it's still stimulated by glucose. It just needs a higher concentration to reach the same enzymatic activity as, let's say, hexokinase 1, as in sort of like as sort of extrapolating hexokinase 4 to hexokinase. You need like like twice as much or 10 times as much concentration. Just
Yeah.
1 second.
Outside.
Yeah, no
Okay.
problem. Uh, Uh, okay, and well, so here you have muscle and liver hexokinase, and just to reinforce the concept, so you have hexokinase 4 in the liver, and you let me just double check that. Muscle, uh, hexokinase in the liver. Yes, hexokinase 4 in the liver. And so, and you have hexokinase 1 in the muscle. Okay. And as they're showing here, okay, the as they say, the liver responds directly to increasing blood glucose with increased turnover. As in, like, as they're showing here for the hexokinase 1 with the muscle. Okay, let's just say it doesn't respond. It doesn't respond much, as in it doesn't take much to turn it on, right?
Right.
As in you need just a little bit. As in, like, to think of the logic of like why it would be regulated in this way, or why it evolved to regulate this way, as in why would liver be more responsive to glucose, or at least you're covering a range of glucose concentrations, as opposed to hexokinase 1. It's just like you add a little bit and you're like pretty much like maximally turned on.
Yeah.
Are you able to like elaborate of like, oh, well, why would that be in terms of like liver versus muscle, and like the demands of the tissues and all that stuff?
So muscle is much more energetically demanding, so it needs to more readily use that glucose for energy, whereas the liver typically will be more for spillover and to potentially uh, you know, so what I'm looking for, move into glycogen stores.
Yeah, yeah, exact. As in perfect. Like since muscle is way more metabolically active as opposed to like liver, it's like, well, yeah, if we just have a little bit of glucose there present, like we generally want to like maximally carry out glycolysis in the muscle, because it's like way more metabolically active as opposed to liver. Okay, great. Regulation of hexokinase 4. Yes, as you I believe you correctly pointed out, when fructose 6-phosphate is high, like that sequesters hexokinase 4. So essentially, fructose 6-phosphate essentially inhibits or deactivates hexokinase. And let's see, and yeah, high glucose weakens the enzyme regulator interaction, encouraging cytosolic localization. Which also makes sense in terms of, well, if you have glucose there, then like we want to activate hexokinase, generally speaking.
Yep.
Okay, and so yeah, you have this whole circuit in terms of, well, when hexokinase is sequestered, like how do you get it unsequestered? As in here, what they're showing here is hexokinase, and you have your regular regulatory protein in the nucleus. Okay, well, how do we get it out of the nucleus? Okay, and like get it to do its thing. And they're going to go into pretty much like how when it's sequestered in the nucleus with that regulatory protein, it's like locked away. It can't do its thing. And then how do you break that interaction between hexokinase and the regulatory protein? And that's where the fructose 6-phosphate comes in, where in this little crude schematic, you pretty much, do you happen to recall? It's okay if you don't, but do you happen to recall like what this NLS stands for?
That is... Nope.
That is your nuclear localization signal.
Okay.
And because like this is so it's not obvious here with this crude schematic, but this is all in the nucleus, as in like
right.
We are in complex here, scrolling up. Yeah, we're in complex here. Hexokinase 4, regulatory protein, like we're locked away in a cage, in this case the cage being the nucleus, or you're doubly in a cage, as in you have a protein stuck to hexokinase 4, and then you're also inside the nucleus. And so how do we break the interaction between hexokinase 4 and the regulatory protein, and how do we release hexokinase 4 from its cage, or release it from the nucleus? Okay, and so that's where the fructose 6, and that's why you have this nuclear localization signal on the regulatory protein, as in it's a nucleus, so it needs something to signify or to give it its passport of like, hey, you at least for now, you belong in the nucleus. But then that nuclear localization signal should change. Okay, so pretty much once your fructose 6-phosphate binds, okay, I want to make sure that I'm going sequentially in this crude schematic of like, you have your hexokinase 4, you have your regulatory protein, and then your fructose 6-phosphate, okay, comes along, and obviously there's going to be like a threshold of like once you reach sufficient concentration, okay, then you will have an interaction between the fructose 6-phosphate and the regulatory protein, okay, and then that's probably going to promote the interaction, as they're showing here, between hexokinase and the regulatory protein, okay. So you have your nuclear localization signal, as in that's going to drag it to the nucleus, okay, where pretty much we're sort of like reeling in something, so to speak, of like your regulatory protein interacts with fructose 6-phosphate, and then your regulatory protein has a nuclear localization signal on it. So when it forms this complex with hexokinase, okay, well then that's going to sequester it in the nucleus. because it has this nuclear localization signal on it. And so yes, to the nucleus we go, as in pretty much once we're in this full complex of fructose 6-phosphate binding to your regulatory protein. And then almost certainly what's occurring is like the binding of fructose 6-phosphate is like exposing, not only exposing your nuclear localization signal, but is also promoting the interaction between your regulatory protein and your hexokinase. And so when they form this complex, and by virtue of the fact that your regulatory protein has your nuclear localization signal on it, then you're dragged into the nucleus.
Okay.
1 second. Okay, well, yeah, now we're in the nucleus. Okay, so now we're sequestered away. Hexokinase can't do its thing; it's in the nucleus. But then at some point, this interaction or this complex, will be disrupted or broken. And that's where glucose comes along. Where, as you mentioned before, glucose stimulates or activates hexokinase, and so that's probably going to, you are now changing. You're going from a nuclear localization signal to a nuclear export signal. Okay, as upon the binding of glucose, okay, and your nuclear export signal is on your hexokinase, so that's going to be released from the nucleus. Then it can go carry out its catalytic activity, or convert glucose to glucose 6-phosphate.
Right.
I guess that's the end of the animation.
So and it's staying in the nucleus because of the fructose 6-phosphate.
It should be this complex of the fructose 6-phosphate and the regulatory protein. It should be all three of these things to get, because
right.
As they're showing here in the diagram, okay, obviously it's a crude diagram. These are simplified figures, but
yeah.
Let's scroll back. Yeah. So it's a little bit. It's actually a lot subtle, but. So in the absence of your fructose 6-phosphate, as your fructose 6-phosphate is not stuck or is not bound, it's not bound to your regulatory protein, and so in this very subtle figure here, okay, the little triangle shape on the hexokinase and the regulatory protein, like they can't interface, they can't interact. Okay?
Right.
As in your triangle on the regulatory protein is like truncated. Okay.
Okay.
But then the fructose 6-phosphate reaches your threshold concentration, and then the fructose 6-phosphate binds to your regulatory protein. And then, it's very subtle of like, well, now you're a full triangle.
Right.
And so now I can interact with hexokinase. Or that's why I say like the binding of fructose 6-phosphate is likely inducing a conformational change, and they're trying to show it with the formation of the triangle of like, well, now
right.
I can actually, in the absence of the fructose 6-phosphate, like you can have your regulatory protein and your hexokinase, like they can be there, like in close proximity next to each other. They're not going to interact, as in you need that fructose 6-phosphate that forms the full triangle, and now you're promoting that interaction between hexokinase and the regulatory protein.
Okay.
1 second. So yeah, it's pretty much all those three things. That's why, as they're showing here, when your fructose 6-phosphate is bound to your regulatory protein, you have the formation of the full triangle that can now interface with the triangular shape on the hexokinase. And that's why in the what's likely occurring with the glucose is, again, very subtle. Like, well, here, okay, glucose outside the nucleus gets inside the nucleus, but now the glucose binds to the hexokinase 4, and that that's going to induce a conformational change on the hexokinase. If you can, it's subtle, but the triangle shape on the hexokinase is no longer there. And so that's like disrupting the interaction between the regulatory protein on hexokinase 4, where there's probably two things going. Well, there's for sure one thing going on in terms of a conformational change on the hexokinase 4, such that you are disrupting the interaction with the regulatory protein. And then two, you are likely exposing the nuclear export signal on the hexokinase 4.
Okay.
And so that's how you are, well, one, that's how you're forming the interaction between the regulatory protein and the hexokinase. And that's how you are breaking the interaction between the hexokinase and the regulatory protein.
Right. Yeah, that makes more sense. I definitely missed the, somewhat subtle crude diagrams.
I would say very subtle, like
yeah,
which I would say totally like understandable that anybody would miss it. Or if you're not used to seeing things depicted like, well, even I'm not necessarily used to seeing things depicted like this, of just simple shapes made in PowerPoint or something. But like, yeah, it is very subtle, but that is the message they're trying to convey. Triangle to triangle on the two proteins, but in the that's with the presence of fructose 6-phosphate. But then in the absence of fructose 6-phosphate, when your regulatory protein isn't bound to fructose 6-phosphate, it's like, oh, triangle on hexokinase and like a blunt triangle on the regulatory protein. Very, very subtle and hard to clue into, for sure. But yes, so definitely key thing there is like you need the three things. Okay, you need the fructose 6-phosphate on the regulatory protein to form the complex with the hexokinase inside the nucleus. 1 second. One moment. Okay, well, we've seen this before, so, and as you reiterated, glycolysis and gluconeogenesis, the key steps. Okay, now we're getting into more allosteric regulation of, PFK1 or phosphofructokinase 1, and yes, you have these allosteric regulators. And you have activators and inhibitors, where, if you're able to, as best you can, of like, well, why would, are you able to sort of like extrapolate the logic behind this in terms of like citrate inhibits, ATP inhibits, fructose 2,6-bisphosphate activates, AMP ADP activate.
Yeah, so ATP and citrate are, basically showing that you have enough energy that you don't need to continue through glycolysis to produce more energy, whereas things like AMP, ADP, and fructose 2,6-biphosphate are showing a lack of energy. Fructose 2,6-biphosphate, though.
That was way more tricky. There's the whole I should have just said this.
Not exactly, but yeah, like I know that it is its strongest regulator, but. I guess it's a caveat.
But everything you said about the ATP, AMP, citrate, like perfect, wonderful. Again, goes back to that concept. At least I like the concept of energy charge of like citrate and ATP are signaling, high energy charge, as in we have essentially excess ATP or citrate. So let's downregulate PFK1 and thus downregulate glycolysis. We don't need to break down more glucose. And then yes, AMP, you're signaling, or AMP ADP, you're signaling low energy charge. As in, oh, we need to produce more energy, so let's start upregulating glycolysis. Perfect. And yes, I'm glad that they include this, the glucose 6-phosphate, as we've seen now going through all the slides, although I don't expect you to remember, like it has multiple fates in the cell and can be repurposed for many things, as we'll see. And yes, and PFK1, that's also, I'm glad they're using that term, it's also a committed step. It is irreversible. Okay. Oh, and yes, ATP, as you just went through. But yeah, this is an allosteric site, not the active site. Relative inhibition by, yes, ATP. Okay, they're just going through all the same thing. And yeah, as you correctly said, the fructose 2,6-bisphosphate is the strong activator. And like, yeah, there's a little, let's just say it's a little bit more complicated than just the ATP, ADP, and AMP.
Yeah.
In terms of like, well, we'll get to it, but
doesn't as much have to do with energy charge.
Yes, exactly. A little bit more indirect. And like there's sort of well, if I remember correctly, but we'll get to it. Of like there's that dual role between fructose 2,6-bisphosphate and how it activates either glycolysis or gluconeogenesis and like deactivates. So basically, it has reciprocal effects, as in it's activating one as it's deactivating the other, if memory serves me correctly. Okay, yeah, and they're just showing here the graphs that we've seen before in terms of energy charge and the concentration of ATP. Like, yeah, as you just went through and explicitly said perfectly, like under low concentrations of ATP, you are going to be stimulating phosphofructokinase 1 activity because you want to upregulate glycolysis. Okay. Oh, this is the review we just went through. Go through that quickly. All this stuff. Oh, it actually, when I scroll through it pretty quickly, it actually animates pretty decently.
Oh, yeah.
Okay, pyruvate kinase. Oh, am I missing something? Okay. But yes, well, I thought they were going to dive right into the fructose 2,6-bisphosphate. But yeah, the pyruvate kinase. Yes, inhibited by ATP, which again makes sense. All under the concept of energy charge. Or like, yeah, if you have excess ATP, well, then we can downregulate the last step of glycolysis, or pyruvate kinase. And yeah, and it's an allosteric regulator of pyruvate kinase. And to hopefully tie it into like just this concept here of like promoting interaction and, let's say, affinity, of like, to put it in terms of like affinity, so to speak, of going back to this of like, okay, in the absence of fructose 6-phosphate, okay, hexokinase 4 and the regulatory protein, they have low affinity for each other, where basically they're not, well, one, they're not interacting, and they're not interacting because probably the amino acid residues, okay, that are there present to promote the interaction are not exposed. You kind of think of it as in, let's say there's a hydrogen bond that holds these two together when they are interacting. But then in the absence of fructose 6-phosphate, okay, the amino acid residues that are involved in that hydrogen bonding, they're not exposed. They're not in the correct orientation. Okay.
Wouldn't that be like kind of like referring back to the
yes, I am.
tyrosine kinase
exactly.
Dimerization. Yes. Yeah, exactly.
Perfect. Yeah, that's what I was trying to relate it to. Of like, yeah, in that tyrosine kinase, when they're not dimerized, okay, as in you don't have insulin bound, right? Then we don't want to autophosphorylate, right? So what happens is you have that aspartate residue, and you have a hydrogen bond between like aspartate, like 1152 or something, and then between one of the tyrosine residues, and that hydrogen bond basically blocks off the active site, so that we can't autophosphorylate. As in the presence of that hydrogen bond, the active site of the tyrosine kinase like has low affinity for the site of phosphorylation. And then yes, in the absence of fructose 6-phosphate, in the absence of fructose 6-phosphate, then hexokinase 4 and the regulatory protein have low affinity for each other because, like, yeah, those residues are likely not in the correct orientation or just, like, not exposed so that you can have the interaction occur. And then, yes, once fructose 6-phosphate comes along, there's probably almost certainly a conformational change where now those amino acid residues are now exposed or in the correct, or at least then on the regulatory protein, there's a conformational change on the regulatory protein, since that's what's only changing when fructose 6-phosphate binds. And so that promotes the affinity between hexokinase 4 as a result of a conformational change on the regulatory protein as a result of fructose 6-phosphate binding.
Right.
So essentially, fructose 6-phosphate binding is promoting the affinity between hexokinase and the regulatory protein. And then the glucose, when it binds to hexokinase, it's decreasing the affinity between hexokinase 4 and the regulatory protein. 1 second. I want to just reply to something.
Yep.
So in the case of pyruvate kinase, sorry, scroll too far. Getting back to this of so ATP, that's an allosteric regulator. Okay, so it's binding at a different site on pyruvate kinase, and as they're saying, is decreasing its affinity for phosphoenol pyruvate, as a result of the ATP binding to the allosteric site, and almost certainly resulting in a conformational change, okay, that's going to affect the affinity for the phosphoenol pyruvate. And then, once that ATP is removed, or it's a much lower concentration, it's oh, I'm going to adopt a different conformation, or I will undergo a conformational change, and that's going to promote the affinity for phosphoenol pyruvate. And that's all going to be driven by those non-covalent interactions. Okay, yes. Not a surprise.
Because I think fructose 1,6-biphosphate is the only positive regulator of pyruvate kinase.
Yeah, that's what it looks like. It's, I guess maybe a little bit sad that they don't cover why. So they mentioned alanine, which is great. But I wish that they mentioned why alanine, of all things, would negatively regulate pyruvate kinase. But yes, all of this makes sense in terms of acetyl-CoA and long-chain fatty acids will inhibit pyruvate kinase. If you want to expand on the logic there of why these things would inhibit pyruvate kinase.
Just because, they are, again, more sources of energy, and breaking down those will generate more ATP.
Yeah, perfect. Yes, acetyl-CoA.
Well, acetyl-CoA is the result of pyruvate.
That's true too. Yes, and yeah, all of these ultimately serve the purpose for making ATP. And so if we have an excess of any of these things, then it's well, we're not in dire need to produce ATP. Great. Oh, here she works in why you should do research.
Yeah, that was just she had one of her research students come.
Her grad students, yeah. It's all common. Okay, yes, gluconeogenesis, the opposite, and essentially, not all, but many of the reverse themes that we just went through apply here. Of if low ATP, or I guess I'll be more specific, if high AMP or ADP stimulate or activate glycolysis, then they're almost certainly going to have a reverse effect on gluconeogenesis, and vice versa. Okay, and yes, so this is making glucose, and you already went through all the steps of the differences in terms of pyruvate to oxaloacetate by pyruvate carboxylase, and then oxaloacetate to phosphoenol pyruvate to phosphoenolpyruvate, I think should be carboxylase or carboxykinase.
Carboxykinase,
Yeah, you are exactly right. Okay, yes, precursors. So, maybe not obviously, but hopefully somewhat intuitively, like, glucose 6-phosphate. Let's say a starting point, okay? And this is a nice example of how glucose 6-phosphate can be repurposed to, like, many things, okay? But let's see, scroll down. Oh, this is all stuff that we just went through. Yeah, I don't think I need to go through this because you went through this exactly.
Yeah.
Except for the malate dehydrogenase, it's probably a good thing. Yeah, almost certainly a good thing to review.
Yeah.
There's a nicer figure here somewhere. Or maybe not.
Because no, that's the main way that it basically passes through the mitochondrial membrane to transfer that NADH.
Exactly. Where we want to, well, we're going through gluconeogenesis, right? So we're going from pyruvate to oxaloacetate to phosphoenolpyruvate. Okay, so we need to, like, get things, out of the mitochondria. Okay, and, like, yeah, that malate is like a key player in terms of getting NADH outside of the mitochondria. And you have, I think, as they say, you have two forms of malate dehydrogenase. You have cytosolic malate dehydrogenase, and you have mitochondrial malate dehydrogenase. But they essentially convert opposing reactions, or what one does in the forward, the other does in the backwards reaction. As you can see, of like the mitochondrial malate dehydrogenase, it's from oxaloacetate to malate, and then the cytosolic malate dehydrogenase, it's the reverse of malate to oxaloacetate. And then, like, you have reverse effects of NADH and NAD+, as in, like, in the mitochondria, you are consuming NADH, and then outside the mitochondria, you are regenerating your NADH. Let's see, what else did they say? Yeah, gluconeogenesis consumes NADH. And so, like, we need some sort of source of NADH. And so where's our source of NADH going to come from? This is one of our primary sources, not the only one, but, like, yeah, pretty much that's the link there. Let's see. Okay, great. 1 second.
Show me the money.
All right. Onwards and upwards. Okay, yeah. This is still the same steps. Glucose 6. Yeah, you already went through this. Glucose 6-phosphatase. Okay, yeah. Reciprocally regulated. Okay, yeah, great. Now I want to get into the fructose 2,6-bisphosphate. And so, yeah, you have your reciprocal aspects here, and your complex circuitry, although all the rest I would say makes sense in terms of you have ADP, AMP, they activate phosphofructokinase 1 on the left. And so, yeah, not a surprise that they inhibit phosphofructose bisphosphate 1 on the right. And pretty much anywhere that you see, like, a plus in PFK1, it's like a minus in phosphofructose bisphosphatase 1.
Yep.
Jeez, I need to make sure I'm on point with my phosphates and phosphatases, because
Oh, they're so close. It's like it ends up a tongue twister constantly.
yeah, not fun. But it is important. There is definitely a difference between a bisphosphate and a bisphosphatase.
Yeah.
Absolutely. Unfortunately. Okay, yes, but yes, there is this aspect as shown here nicely in bold of like a fructose 2,6 bisphosphate, okay, where it's activating on one hand and inhibiting on the other, or plus minus, as in fructose 2,6 bisphosphate activates phosphofructokinase 1 and inhibits phosphofructose bisphosphatase 1. So how does it do that? Or there's some, yeah, these nice graphs here. In terms of, okay, getting back to Km or K0.5, you know, roughly the same thing. In terms of, yes, they're showing here nicely that, let's see, fructose 2,6 bisphosphate greatly increases phosphofructokinase 1 activity. Yes, has low affinity for fructose 6 in the absence of fructose 2, in the presence. Okay, yeah. So they're showing how, okay, fructose 2,6 bisphosphate and how based off of the presence or absence of fructose 2,6 bisphosphate, that alters the activity of phosphofructokinase 1. Okay, well, where we've seen these graphs before, and basically one of your main messages is that in the presence of fructose 2,6 bisphosphate, okay, you are increasing, as they say, the affinity for PFK1 for its substrate. Where you're and its substrate is fructose 6 phosphate there. So on the x-axis of your graph, you have your substrate. You have your substrate for fructose, phosphofructokinase 1. Okay, and then what they're showing is in the presence of fructose 2,6 bisphosphate, it requires a low affinity, a lower concentration of your fructose 6 phosphate to generate the same activity. As I mentioned before, I guess follow or keep track of the dotted line of how that's changing in the presence or absence of fructose 2,6 bisphosphate. Where in the absence, okay, you basically need exactly, according to this, 2 millimolars of fructose 6 phosphate to get to 50% activity of PFK1. As opposed to in the presence of fructose 2,6 bisphosphate, you need much less, as in less than 0.1. This looks like a 0.05 or close to 0.05. As in you just need a little bit of the fructose 6 phosphate to get to 50% activity. So that would be your main message here of how fructose 2,6 bisphosphate alters or regulates phosphofructokinase 1 activity. So basically, presence stimulates, absence, inhibits. Although again, it's not full inhibition. You can still turn it on. It just requires a much greater concentration of the fructose 6 phosphate.
Right.
Okay, and then we're going to see sort of a reciprocal thing with the fructose 1,6 bisphosphatase activity. Not fun to have fructose 2,6 bisphosphate and fructose 1,6 bisphosphatase on the same slide. But that's the reality that we gotta live with and deal with. But yes, so maybe I'll try to get both graphs on the screen. Yes, where basically comparing like with like, as in fructose 2,6 bisphosphate in the presence for both. Well, in the presence, when it comes to PFK1, that stimulates, as opposed to in the presence, you're not inhibiting, but you're definitely not stimulating. Okay, in terms of like you require pretty much a much higher concentration of the substrate of fructose 1,6 bisphosphate to reach 50% activity. And then when you compare like with like, in terms of absence of fructose 2,6 bisphosphate, Well, with PFK1, you're like inhibiting, you're definitely not stimulating, as opposed to in the absence of fructose 2,6 bisphosphate, you are explicitly stimulating FBP1 or fructose bisphosphatase 1. Okay, yeah, those are your main messages, but basically, like, probably use this as your anchor. If you just basically remember this on that fructose bisphosphatase
Yep.
is pretty much the reverse of that. Of like,
Yeah.
whatever you're seeing here is the opposite for fructose bisphosphatase 1.
Yep.
Okay. This is all review, seen before. This is all literally what we just went through. Okay. So still on the same theme of, although this is phosphofructokinase 2 and fructose bisphosphatase 2 regulation, where, let's see, it's bifunctional, where reciprocally regulated. Okay, yeah, same thing. Let me just see. It's not insulin. Okay, pentose phosphate pathway. Great. All right. So, well, you have your opposing pathways on the left and the right. You have insulin, okay, which results in the uptake of glucose from the bloodstream, and you have glucagon, which promotes glucose release into the bloodstream, okay. And so you have your two enzymes, okay. You have your phosphoprotein phosphatase, and you have your CAMP-dependent protein kinase A, or PKA, okay. And so you have your PFK2 and FBPase2, okay, on the same enzyme. As they say, it should be a bifunctional enzyme. Let me just bifunctional enzyme. And so yes, they are, as they say, reciprocally regulated, where when one is on, the other is off, and when the other is on, the other is off. As in, when PFK2 is active, FBPase2 is inactive, and vice versa. And this is all regulated essentially by phosphorylation, where you have your kinase, your CAMP-dependent kinase, or PKA, that will phosphorylate the enzyme, and that essentially will switch it. That will switch on the FBPase2 domain of the enzyme. That would be like your critical point to remember. Basically, dephosphorylated PFK2 is active. When you're phosphorylated, your FBP, your phosphatase 2 is active. And then, yes, glucagon, when you get your signal from glucagon, that promotes phosphorylation of the enzyme by PKA. And then, like, yes, when you have insulin, that promotes the dephosphorylation of the enzyme. As usual, a complex circuit. Okay. Let's see. Pentose phosphate pathway. Yeah, well, so, just to reiterate and reinforce the point, like, even though you don't cover much of it, you cover the main points. So, like, what's the whole point of the pentose phosphate pathway, or what is it largely responsible for?
Creation of NADPH and nucleotides.
Or nucleotide precursors.
Nucleotide precursors, yes.
Yeah. As in, like, your ribose, phosphate. Yes, exactly.
Yes.
Perfect. And definitely NADPH. And again, just to reinforce the concept, NADH as opposed to NADPH. Obviously, one contains phosphate, the other one doesn't. But in terms of, metabolic responsibilities, like NADH largely responsible for, you know, this in metabolism, and NADPH largely responsible for this in metabolism.
NADH largely responsible for catabolism, NADPH largely responsible for anabolism.
Yep, perfect. Wonderful. Okay, yes. Yeah, gluconeogenesis occurs in the liver, well, major site of gluconeogenesis. And again, to reinforce the concept, where we covered briefly, you know, that question about gluconeogenesis in the liver versus gluconeogenesis in like the muscle or the brain, as in like you essentially don't find gluconeogenesis in the brain or the muscle all that much. And like why would that be sort of metabolically, as in like why did we evolve having the liver as the major site of gluconeogenesis as opposed to like, you know, the brain or the muscle?
Because again, the muscle and brain are much more metabolically active and require more of that ATP on like rapid demand, whereas
Yeah.
the liver is kind of not sitting idle, but it doesn't require all that ATP immediately all the time, and it
Yes.
is largely a store of glycogen a lot of the time.
Yeah, perfect. Right on the bullseye. Okay, regulation of pyruvate kinase. Okay, great. You know, last step of glycolysis. Let's go zoom in. Let me just get it centered. Okay. So, yes, maybe not surprisingly, you know, phosphorylation plays a role in regulating pyruvate kinase, where yes, in your dephosphorylated form, then your pyruvate kinase is active, and so yes, this is mediated by protein kinase A, where yes, you are getting your signal from glucagon, okay, and then that will activate protein kinase A, and then that will phosphorylate pyruvate kinase, and like then you are inactive. Okay, and like let's say this should make metabolic sense in terms of so pyruvate kinase, just to reinforce the point of like pyruvate kinase is responsible for what step in glycolysis?
That's changing phosphoenolpyruvate into pyruvate.
Yes, exactly.
Yes, and which is to generate energy, and obviously glucagon is kind of like working in the reverse.
Yes, exactly. So to speak, of like, yeah, glucagon. We want to. The signal is like to stimulate release of glucose into the blood, right?
Exactly.
And
Yeah.
therefore that means that would seem that we're under low glucose. And so if we're under low glucose, then it's like we don't really have we can't really carry out glycolysis, or we don't really want to carry out glycolysis. Because we want to carry out glycolysis, well, that means we want to take up glucose from the bloodstream, right? And that's where insulin would come into play. If we want to carry out glycolysis, that would be uptake of glucose from the bloodstream. Then here we're literally saying the opposite, you know, of like, well, put it in the bloodstream. Okay, and I think everything else sort of like hopefully also makes sense in terms of energy charge and all that stuff. ADP, ATP, all the same things. Acetyl-CoA, fatty acids. I think it's all the same stuff that we've seen before. Yeah, the malate, or rather, the malate dehydrogenase, and getting NADH outside of the mitochondria, especially if we want to carry out gluconeogenesis. NADH is needed. Perfect. This is like, I think, just an overall summary slide, which is great. Okay, yes, pentose phosphate pathway. Thankfully, you don't spend that much time on it. I think just like 4 or 5, yeah, 4 or 5 slides. Thankfully. So, but yeah, we already very briefly reviewed it. But yes, pentose phosphate pathway. Essentially, we just largely want to generate NADPH for biosynthetic purposes, like we'll see for fatty acid synthesis. And then yes, we want to make precursors for nucleotide synthesis. Generally speaking, that ribose phosphate. Yes, reductive biosynthesis and ribose 5-phosphate. Okay, yeah, regen. I don't think that's super important. Okay. I don't think you really need to know this pathway. Probably just the endpoints there. In terms of ultimately, we want to generate ribose 5-phosphate because those are that is a main precursor to the nucleotides, and then yes, also main output NADPH for sure. And the rest is just I would say annoying stuff in shuffling a bunch of carbons around, which is not overly fun. I mean, I don't know if this is important or not.
She didn't spend too much time on it, so
yeah,
I wouldn't worry about it too much.
I wouldn't think so, and I would hope not. As in, at least based off of number of slides and like overall, like I'm hoping the main focus is regulation of glycolysis and gluconeogenesis, and fatty acid stuff, synthesis and oxidation.
I think again, like we kind of mentioned, like it's mostly just largely to mention that it's to produce NADPH and the nucleotide precursors.
Yeah, I think that's fair. Okay, yeah, regulation of pentose phosphate pathway. Okay. Okay, glycogen metabolism, where we're getting into glycogen phosphorylase and glycogen synthase, which we have seen before. We saw it a little bit, with signal transduction and all that stuff. And just to reinforce the point, because like all these enzyme names are confusing, but yeah, glycogen synthase responsible for the synthesis of glycogen, and then your glycogen phosphorylase responsible for the breakdown of glycogen. And yeah, your glycogen phosphorylase should have essentially 2 forms. You have glycogen phosphorylase A, and you have glycogen phosphorylase B. And your A form should be your active form. At least that's what I make the association as. Glycogen phosphorylase A, that is active, and that allows you to break down glycogen into its respective glucose monosaccharides. Okay, but yes, how do we make glycogen? Glycogen via glycogen synthase. And so yes, you have your mutase, where ultimately, since we are basically connecting a bunch of glucose units, as usual, as I like to say, we need to use an activated form of glucose. And UDP glucose is definitely an activated form of glucose that we see all over the place in biochemistry. But before we make our UDP glucose, we need glucose 1-phosphate, and that's where your mutase comes in, where all we're essentially doing is changing where the position of the phosphate is from the 6th position to the 1 position. And then we activate it.
The UDP can bind to the 6th position?
Yes, exactly. Or rather, actually, it should be the 1 position.
Yeah, probably.
Because you're releasing a phosphate. Because we want to activate that site specifically to incorporate.
Yeah, and there you go.
Yes.
You're on the 1 position.
And glycogen is branched, and depending on where you are in the branching, like the links should be a little bit different. Do they show? Okay, no, they don't show it. Then maybe you don't need to know it. But yeah, your main chain is definitely carbon 1 to carbon 4.
Yeah.
And then I think on the branch, if I remember correctly, it should be like carbon 1 to 6. But I don't think you really need to know that all that much. But there's definitely a main chain, and glycogen is a little bit branched. And how it is on the branch is a little bit different, or the linkages are different. But yes, ultimately all that we're doing is we are starting with glucose 6-phosphate. We are converting it to glucose 1-phosphate by that mutase. And then we are activating our glucose, and then we are incorporating it into our growing glycogen chain. All right. Yes. And then in the opposite, so this is glycogen synthase reaction, and then its opposite is the glycogen phosphorylase reaction. So to break it down, and yes, I mean, you're just releasing the glucose unit, nothing overly fancy. But it is, I guess, pretty critical to keep in mind that going back to make glycogen, we start with glucose 6-phosphate, and it's converted to glucose 1-phosphate by the mutase, and then it's activated. Okay, and so, but to get into the into glycolysis, you want to enter glycolysis as glucose 6-phosphate. As in, this glucose 1-phosphate needs to be converted back into glucose 6-phosphate in order to be usable in glycolysis. And then, so the mutase comes back in again and catalyzes the reverse reaction.
And it's the same.
Yes, it should be the exact same mutase. Yes.
Okay.
Where it should be a reversible reaction.
Yeah.
Almost. Well,
like that should be a bidirectional arrow.
Yes. Given the fact, and also given the fact that it's named the same thing, like hopefully you're maybe getting an appreciation of even though it's confusing, we name these things very specifically. Of like, if we're making a distinction between glycogen phosphorylase A and glycogen phosphorylase B and all that stuff, then if they're using the same name here, phosphoglucomutase, in the phosphorylase reaction, it should be. And I'm like 99% sure that it's the same phosphoglucomutase in the glycogen step as well. Otherwise, well, it gets even more confusing if we're calling two different things the exact same name. Okay. Oh, this is all review for the pentose phosphate pathway. Okay. Glycogen metabolism. Yes, just went through this. Great. Okay, yes. Regulation of glycogen metabolism. And like, yes, there are many things that regulate glycogen synthase, but ultimately, as they're showing here, you have your main regulators, definitely your glycogen synthase kinase 3, which since it's the kinase, going back to that circuitry from that signal transduction pathway of the kinase, when the glycogen synthase kinase phosphorylates glycogen synthase, that inactivates it. And then and it's one of the main. It's actually a little bit sort of paradoxical, If you just show this, typically when we show plus signs in biochemistry, that most people intuitively think like activation, right? Or like, well, plus. But the column title is degree of inactivation, so it's like plus of inactivation. So it's a little bit say not the best, but like yes, that's so the glycogen synthase is inactivating.
It's positive, negative.
Yeah, exactly. You can't get any more confusing than that. But yeah, we're doing a lot of inactivation, plus of inactivation, or plus of minus. Very strange, but it is what it is. I'm hoping that maybe in the 8th edition they changed this convention, but it's a little bit counterintuitive. But yes, that is important to keep in mind. And like, yes, the glycogen synthase kinase 3 is like one of the key regulators, since comparing it to the other ones where they just have a singular plus sign. The glycogen synthase is like, it's a major deactivator or inhibitor when it phosphorylates glycogen synthase. And yeah, not a surprise as they're showing here at the top. As in you have multiple phosphorylation sites on glycogen synthase. Okay, you probably don't need to know any of them, but you probably do need to know that there are multiple phosphorylation sites on glycogen synthase.
Right.
And well, they do tell you here in this table of what are the exact sites of glycogen synthase kinase 3. It's at site 3A, 3B, 3C, which will be right there, 3A, 3B, 3C. But I mean, I'm hoping you don't need to know that level of detail. Okay, so yes, glycogen phosphorylase. So here it's like a reciprocal. So here we're talking about the breakdown of glycogen. Okay, so as opposed to what we just saw with glycogen synthase, okay, glycogen synthase is inhibited when it's phosphorylated, especially when it's phosphorylated by glycogen synthase kinase 3. But then here for glycogen phosphorylase, it is, as it says, it's activated by phosphorylation, and its active form should be, yeah, as shown there, phosphorylase A, and I make the association with A for active. Whether that they named it that intentionally for that, I don't know. But at least that's how I try to anchor myself. And so yes, not a surprise. You have a kinase and you have a phosphatase. Okay, and so you have phosphorylase. You know, very at least confusing to me. Of phosphorylase B kinase, so it's going to phosphorylate phosphorylase B, that converts it into phosphorylase A, that is now active. Okay, so we're basically, you know, we are converting phosphorylase B into phosphorylase A by phosphorylation by phosphorylase B kinase. Yeah, that's very confusing. But let's say, so glucagon, as in like, well, what they're showing here is glucagon, the signal from glucagon in the liver stimulates this. Well, what does it stimulate? It stimulates phosphorylase A, ultimately, where if we go through the circuit. So glucagon, okay, the signal from glucagon stimulates or activates phosphorylase B kinase. What does phosphorylase B kinase do? It phosphorylates phosphorylase B and converts it into A, as in we're now active. As in basically, the signal from glucagon is ultimately leading to phosphorylase A being active. Okay, well,
right.
Does that make sense? The answer is yes, in terms of, well, what is the point of glucagon? Glucagon is to get glucose into the bloodstream, right? We want to, as opposed to insulin, take up glucose from the bloodstream. Glucagon, get glucose into the bloodstream. Well, right. How do we want to do that? Well, pretty much one of the main ways to do that is to break down glycogen, okay, and get it into the bloodstream. And so that
active phosphorylase A is, yeah, okay, that's a breakdown.
Yes, yeah, exactly. Yes. Again, very confusing, but yeah. But the glycogen synthase, that's when you're making glycogen. I try to anchor it just like with fatty acid synthase. That's responsible for making fatty acids. We are synthesizing fatty acids. Glycogen synthase, we are synthesizing glycogen. And so the phosphorylase, we are breaking down glycogen. And so that makes sense in terms of glucagon. We ultimately want to result in getting glucose into the bloodstream. Well, what's one of the primary ways that we can do that is to break down glycogen and release it into the bloodstream. So that makes sense metabolically. And it also makes sense metabolically in terms of, well, epinephrine. Epinephrine will activate phosphorylase B kinase. In terms of, well, if we're in fight or flight and we need a supply of glucose, well, then we want to get glucose into our bloodstream so we can transport it across the body and get it to, as they're showing here, like muscle and other tissues. that are needed for your fight or flight response. Okay. Okay, they go through it in words here. Is stimulated by PK-mediated phosphorylation. Yes. Yes. So as they say here, the glycogen phosphorylates A dephosphorylates glycogen phosphorylase. So again, it's super confusing. You have a phosphorylase A phosphatase, but you also have phosphorylase A. So what does the phosphorylase A phosphatase? It dephosphorylates phosphorylase A and pretty much turns it off. Or now we are in our inactive form, the phosphorylase B form. Okay. Great. Wonderful. Let's see. Yes, phosphorylates glycogen, okay, we already just went through this. Is a glucose sensor, which makes sense of like what are they showing here of when you have glucose bound, you're in your phosphorylase B form, and remember your B form is in your inactive form. Okay, and that makes sense metabolically, because like what's the point of the phosphorylase when it's turned on is to release is to break down glycogen and release glucose into the bloodstream. Well, we don't want to release glucose into the bloodstream if we already have glucose there. Okay, and so when glucose is bound, as in you have these nice binding sites on the phosphorylase B, that will as in that maintains the phosphorylase B as the phosphorylase B. Like, hey, we don't need to release any more glucose, or we don't need to break down glycogen to release any more glucose.
Right.
And then yes, no surprise here of like insulin. Okay, does it, like, let's say, make sense that insulin will stimulate the phosphatase? Okay. And the answer is should be yes. That does make sense, as in
because it brings more glucose into the cell to bind to phosphorylase B, making it less active.
Yes. Let me just think that through. So insulin is stimulating the phosphatase, right? And what does the phosphatase do? The phosphatase
dephosphorylates the active.
Yes. Yes.
Turn the inactive.
Yes, exactly.
Yeah.
Yes. Perfect. Yeah. And yeah, that makes sense. Okay. But yeah, a lot of this, these are almost basically like logic problems in terms of, A, if A, then B, but if B, then C. Okay. Okay, well, they're just showing here the reciprocal and all the things of, you have glycogen synthase and the glycogen phosphorylase. Where, yes, you have sort of opposing things of when, as I mentioned before, when your glycogen synthase is phosphorylated, you are inactive, but when your phosphorylase is phosphorylated, then you are active. And here, now going back to your glycogen synthase kinase 3, that's carrying out the phosphorylation of your glycogen synthase, and that is inactivating it. But then your phosphatase, does the reverse. Just as your phosphorylase B kinase, turning it on, or converting it from phosphorylase B to now phosphorylase A, that is phosphorylated. And then you have your protein phosphatase. And it seems like, at least based off of this, that your protein phosphatase is like playing the same role in both, as in they have it in the middle there. In terms of the protein phosphatase 1 dephosphorylates glycogen synthase B, and then your protein phosphatase 1 dephosphorylates glycogen phosphorylase A. Okay, great. Regulated by hormones, okay, and going through this logic of as mentioned, epinephrine muscle, as in, epinephrine, our sort of fight or flight hormone. And so does it make sense that you would activate phosphorylase B kinase? Again, the answer is yes, because if you're in fight or flight, then you want to mobilize your energy stores, like your glucose in your liver, in the form of glycogen. So you want to break down glycogen to release it so that, you can fight or flight.
Yeah.
And then yes, your insulin is activating your phosphatase, as in, yeah, you are. If you're activating your protein phosphatase 1, let's say for the glycogen synthase, as in you are favoring the active form of glycogen synthase. And that makes sense in terms of insulin. Well, insulin, you are under high blood glucose conditions. And so if you're under high blood glucose conditions, well, that means we should have some excess glucose. So what do we want to do with that excess glucose? We want to make some glycogen. And so you would favor, or it makes sense that you would activate protein phosphatase 1 that would favor the active form of glycogen synthase. So you can make glycogen with your excess glucose in your bloodstream. Okay, it seems like the same thing. Okay, yeah. A state, or relaxed or tense state, similar to hemoglobin. But nothing really new here, I don't think. Other than, yeah, your A's, your
I think it's just relating it.
Okay. Yes. Hormone regulation of glycogen metabolism in liver. Okay. So, okay, yeah, they're going under low blood sugar to high blood sugar. Well, at least, well, we just went through the high blood sugar, but now they're adding a little bit more detail in terms of, high blood sugar, insulin release, and then, yes, you favor glycogen synthase, as in you have excess glucose to play with. And yeah, you have your protein phosphatase and your GSK3, all the same thing. So insulin release will inhibit GSK3, the kinase 3. So if the kinase 3 is inhibited, then that means you will not be phosphorylating glycogen synthase. And if you're not phosphorylating glycogen synthase, that means glycogen synthase is activated or on, and therefore you can make glycogen. As opposed to, on the flip side, insulin release, high blood sugar, you are going to activate your phosphatase. Well, your phosphatase will favor your glycogen phosphorylase B, which is your inactive form of glycogen phosphorylase. So you will not be breaking down glycogen, since it doesn't make sense to break down glycogen under high blood sugar conditions. And so that all metabolically makes sense. And then the reverse, then, well, pretty much it's going to be the flip side for the low blood sugar. Yes, you favor the breakdown of glycogen by glycogen phosphorylase A when you are low blood sugar, because you have no blood sugar in your bloodstream. So mobilize your glycogen stores in your liver. Okay, yes. But in muscle, it's pretty much, I think, all the same things. Except, low blood sugar, no glucagon release in the muscle. Since, again, you're not really going to be having, as mentioned, you don't really have, major glycogen stores in the muscle, so to speak. In terms of, low blood sugar. Well, okay, hormone regulating hormones. Yes, went through. This is the same thing in figure form. This is, yes, I think all the exact same things that we just went through. Okay, and tissue specific, which we touched upon before, but always good to review and reinforce. Why are muscle and liver metabolism different? Ultimately, as mentioned, as you covered, it's like, well, they have their metabolic demands are different. Like muscle, way more metabolically demanding than liver, generally speaking. And yes, myocytes lack glucagon receptors, as in like you're not really going to be releasing glucose from your muscle like into the blood. Doesn't make any sense because like
Yeah.
just as like as mentioned, yeah, your brain and your muscle don't really carry out gluconeogenesis because like they're not really they have essentially no use for it. As in, if I'm going to make glucose, I'm just going to burn it right away because I'm metabolically demanding. Or rather, like something has gone wrong in terms of like priorities of the organism have gone wrong if muscle and brain are just like making glucose. It's like no, you should be using any glucose that's around as priority. Just as
fasted for a month straight.
where as you should have covered like the brain prefers glucose to run on that is its preferential fuel of choice in your typical healthy human. It can run on ketone bodies, yes, but in the presence of both ketone bodies and glucose, it will significantly favor the glucose, and like pretty much like won't touch the ketone bodies unless it really has to. Yes, and yeah, muscle lacks gluconeogenic enzymes. They just can't do it, and just just wouldn't metabolically make sense. And yes, muscle lacks a key enzyme for glucose export because doesn't make sense for the muscle to release glucose into the bloodstream of like, hey, I'm a muscle cell and I'm just going to I'm going to donate glucose to somewhere else in the body. That just doesn't make sense for a muscle cell to do that. It just wants to use energy, not either export it or give it away or anything like that. Okay, yeah, and muscle uses storage glycogen and glucose for itself. Okay, all of this is review covered. Yeah. Great. I think that's essentially like the end of this lecture. Yeah, this is the end of this lecture. Okay, great.
Perfect.
Fatty acids. Okay, yeah, fatty acid synthesis, fatty acid breakdown.
The fun stuff.
Okay. And yeah, definitely some fun to be had. But just to get the broad concepts across and to reinforce them, like again, fatty acid synthesis, fatty acid breakdown. Again, just go through like the broad reactions, in terms of the 4 steps that occur.
So beta oxidation is oxidation, hydrolysis, oxidation, dialase. And perfect. Then synthesis is condensation, reduction, dehydration, reduction.
Perfect. Bullseye. Yeah. Okay. And yes, no surprise, we have all kinds of fatty acids. Stearic acid, palmitate, linoleic acid, etc., etc. Which they also get confusing. And yes, these are long hydrocarbon chains. And as I like to emphasize, they have reducing power. We're just trying to, when we're making fatty acids and storing them as triacyl glycerols, as we'll see, we have our glycerol scaffolding, and then we have our 3 fatty acid tails. And yes, we are essentially trying to store reducing power to use it for future oxidation, or more specifically, beta oxidation. Okay, yes, yeah, your stearic acid, your oleic acid, and yes, saturated versus unsaturated. I'm sure these are all things that you know, almost certainly. In terms of, unsaturated, that we have at least 1 double bond. And yes, gram per gram, like it's no contest, that triacyl glycerols have way more energy potential than anything else, at least that we eat. amino acids, glycogen, etc., etc. And it all comes down to the fact that the triacylglycerols are heavily reduced. If not, the vast majority of it is reduced. Just long hydrocarbon tails. Okay, yes. This is essentially how we store them as triacylglycerols. Our 3-carbon glycerol, and then we have our fatty acid tails coming off of them. Okay. And yes, you almost certainly know this in terms of how we store our or rather in what types of cells we store our triacylglycerols. We store them as lipid droplets inside adipocytes. But those, as they say, the triacylglycerols and your lipid droplets, they are essentially coated in perilipin for a number of reasons. One, for regulatory purposes, and also again, triacylglycerols, they are hydrophobic, and the cell is an aqueous environment, just as, to reinforce the point, when we're transporting fatty acids, let's say in our blood or from one place in the body to another through circulation. Do you happen to recall, it's okay if you don't, but do you happen to recall the protein, one of the major proteins in the blood that's responsible for transporting fatty acids around?
Yeah, that was albumin?
Yep, perfect, exactly. Serum albumin, major fatty acid transporter, because the cell can't and doesn't want to just release fatty acids in the blood, because let's say glucose. Glucose, we don't have an equivalent in our circulation, our blood, to transport glucose. So glucose in the blood, it's in the blood. It's just essentially floating around, and then when it comes across its transporter, gets inside the cell. Can't do that with fatty acids because they are hydrophobic, and the blood and inside the cell, those are aqueous environments, and so we need something to pretty much store them around, store them and transport them around in. Otherwise, they'll just clump together and literally clog your arteries. Well, that happens anyways for some people, unfortunately. But yes. We have our lipid droplets here, okay, coated in perilipin. And yes, you have this whole signaling cascade of ultimately we want to release our triacylglycerols into circulation and get them to the appropriate cells that need them. And so there's a whole series of events well, this is also a nice sort of overlap in terms of, well, what they're showing here is a glucagon, right? Glucagon binding to a receptor, a glucagon receptor, and undergoing some sort of signaling cascade, right? This looks like a G protein coupled receptor, right? With your adenylocyclase and cyclic AMP as your second messenger, and protein kinase A. But to put it in maybe overall context of, the organism of well, we definitely know that glucagon, as we just covered in the last lecture of glucagon, well, that's also responsible for releasing glucose into the bloodstream, as in that will promote glycogen breakdown from the liver and then getting glucose into the bloodstream. But then as you're seeing here, it's stimulating fatty acids being released from the lipid droplets and into circulation. And so this all makes metabolic sense in terms of, well, under what conditions do we want to release, let's say, glucose into the bloodstream? Well, we're more or less not under starvations necessarily, but when we're not, well, let's say between meals or whatnot, but basically, under extreme conditions, under starvation of we don't have a source of glucose, so we got to release it from our glucose stores in our liver. Yeah. And same thing here. Well, when do we want to release fatty acids? It's when we can, let's say under extreme starvation conditions, it's well, we don't have a source of food, so let's mobilize our fatty acids. And so it's almost well, it is a two-pronged sort of attack in terms of, well, releasing glucose into the bloodstream, and that can be that is a source of energy, and then releasing fatty acids into the bloodstream that are a source of energy. Of the glucose eventually gets converted into acetyl-CoA, and the fatty acids also get converted into acetyl-CoA. And so this is a nice, well, I think a nice example of, again, highlighting that, well, one hormone can have, multiple effects, not just like always one-to-one. Like glucagon only results in the release of glucose into the bloodstream. Okay, and yes, you have your serum albumin right here that transports fatty acids into throughout circulation. But yes, you'll get this whole cascade of events. I think they go through it step by step, one by one. But yes, hormone binds receptor, and yes, when your perilipin gets phosphorylated as they go through, this pretty much results in the release of this CGI protein. I don't know how super critical it is for you to know that. I think you just need to know that perilipin gets phosphorylated, and ultimately that leads to the release of a fatty acid that will, enter the bloodstream and into serum albumin. But I mean, they have all these steps here. Of the CGI activates ATGL, and then that goes on to release the first fatty acid, and then more and more. But these are your overall steps. Yes, enter the bloodstream.
I guess that makes more sense. Like, I don't know if you know anything about how some bodybuilders are using like glucagon.
No, I don't. And I'd have to assume that they're probably injecting it then.
Yeah, like using it basically fasted for pre-cardio, which is interesting.
Yeah, that is. Well, I guess that, yeah. Well, yeah, I mean, if you're a bodybuilder and you're looking to burn every single percentage of body fat, I guess that makes sense in terms of get into the bloodstream, and yes, they'll undergo. Well, hopefully they'll undergo beta oxidation and all that stuff.
Well, and to, I guess, increase that effect is using L-carnitine as an adjunct.
Yeah. Which I understand why, but like, it's probably not actually, really helpful, and they probably don't really actually need the L-carnitine, but it's not hurting, other than maybe in their wallets.
Well, I mean, they don't take it orally either.
Okay, well, yeah, if they're getting, supra physiological, maybe, I don't know.
Oh yeah, like probably in the range of like a gram.
Well, that's a lot, but teach their own.
Yeah.
They have every right to do that, I suppose. But yeah, well, on the topic of carnitine, well, how do if we are going to mobilize our fatty acids, well, how are we going to get them to where they need to go, which is ultimately, well, one, inside the cell, and then, yes, we need to undergo beta oxidation and generate acetyl-CoA in the mitochondria. As you know, and as we covered, yes, we have our carnitine shuttle. That pretty much, well, at least your 14 carbon fatty acids, they absolutely need essentially carnitine to get into the mitochondria. We can technically shorter carbon chain fatty acids. I don't know that I take issue with no transporter, but I think but either way, follow what's there for sure.
Yeah.
I'm not going to question the professor. But yeah, so all that to say is like we were not solely reliant on carnitine to like get fatty acids and the mitochondria. That is for certain.
Right.
Okay, yeah, and we covered it before in terms of the carnitine shuttle. Basically, just across the mitochondrial membranes, inner and outer. And let's see. Yes, you're essentially just creating a fatty acyl carnitine, and then but you have a specific transporter devoted to, like, it's not like it's binding carnitine and just like free floating across the membrane. Like you have pretty much proteins. Do they show it? No, they don't show it. Well, it is here in this figure, but it's not super obvious. At least not. I don't think it's that super obvious. But yes. Starting with, you have starting on the outside, your outer mitochondrial matrix. You're going to have your fatty acid, and then it's going to pretty much not on its own, as in you need a specific transporter to transport your carnitine into the mitochondria.
Right.
And that will be specific for, you know, your acyl carnitine specifically.
Yep.
Okay, and then yes, and then we undergo, well, we'll eventually undergo, you know, beta oxidation. Okay, yes. So now oxidation of fatty acids, okay, which is, yes, within the mitochondria, ultimately to produce acetyl-CoA, because again, that will enter into the citric acid cycle / Krebs cycle, forming your NADH and FADH2, which will go on to the electron transport chain and blah, 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 proton gradient, ATP synthase, and then ATP. And then just to reinforce the concept and the pathway, it's like, okay, we're going to be oxidizing fatty acids. If you recall, they don't necessarily expect you to, but like, you know, how many carbons at a time will we be oxidizing?
It's 2.
Yeah, should be 2, exactly, 2 carbons at a time. And that generally works out nicely because we are mostly dealing with even number triacylglycerols. But as we'll see, you know, there are adaptations to deal with odd number carbons, fatty acids. Okay. And so, yeah, and it's actually depicted pretty nicely here. Of, yes, 2 carbons at a time. What they're showing here of, yeah, breaking off 2 carbons at a time. Or not breaking off, oxidizing, beta oxidizing 2 carbons at a time. And it does occur at a specific end that I just want to make sure, like, I get right. Because I know that I got that. I made that mistake last time. It should not be at the methyl end.
No.
It should be pretty much, yeah, at the acetyl end, or the C double bond O.
Yeah.
Which, yeah, makes sense because that will be your alpha carbon, that carbon attached to the C double bond O, and your beta carbon, beta oxidation, will be the carbon next to it. So it all tracks. I didn't want to make that mistake again. And yes, no surprise, we generate a lot of acetyl-CoA, but even during the process of beta oxidation, we do generate some NADH and some FADH2. As in during the actual oxidation steps, there is the production of NADH and FADH2 built into it. Right. Which I think you should see. Yes. Okay, yeah, great. Yeah, at the carboxyl end of the fatty acid chain. And yeah, it'll keep going until only acetyl-CoA is left. All right. So, and well, actually, maybe just to hopefully show it, I'm going to go to the chapter of fatty acids oxidation, fatty acid metabolism. Yes, great. Whoopsies, scrolling too much. So yes, this is your pathway for oxidation. Like, yeah, pretty much at each oxidation step, there is the production of FADH2. Oopsies. In your first step, oxidation, you generate FADH2, and then in your other oxidation step, you generate NADH, which makes sense. So it might seem counterintuitive in terms of, well, we are oxidizing and generating FADH2, but again, we are talking about redox reactions, as in what's getting oxidized. Yep. What's getting oxidized is the acyl-CoA. What's getting reduced is the FADH2, as in it makes sense that we have reduction and oxidation occurring at the same time, which we will always have occurring. And so, yes, pretty much each round of beta oxidation, you're generating at least one FADH2 and one NADH. But then we also generate the acetyl-CoA, which will result in more NADH and FADH2 from the citric acid cycle. So it works out pretty elegantly. Okay. They pretty much go through, steps 1 is they break it down. It's probably, I guess, it's probably recommended that you, if you can, memorize these. Not fun, like the trans delta 2 enoyl-CoA, where like I would just like probably, if you can, like just commit that to memory as best you can, even though it's not fun. But yeah, the trans delta 2 enoyl-CoA, and then the L3 hydroxyacyl, and all that stuff.
Well, and we can pull up the practice questions too and just see if there's anything similar to that.
Okay. I'm not sure if I have the practice questions.
I did upload them to our shared
folder. Okay. Give me a second.
Yeah, sorry. Sometimes like it notifies you, or like it gives me a option to notify you, and sometimes it doesn't.
Let me extract those. To do sample final. Okay, great.
Yeah, one looks like practice short answer or written response questions, and one is like a full practice test, it looks like. And then the third one is just the answers to that.
Great. Wonderful. Okay, I see. Yeah, there's sample final and potential final exam questions.
I always find potential seems like probable.
Almost certainly.
At least I hope so, because I have had, say, my statistics class in year 1. A lot of people failed because the final was substantially harder than the practice. Provided that was like basically our midterm practice exams were nearly identical. So it was like, as long as you can do this, you can do the final. But it was like, or the midterm, but then the final was like a complete different thing.
Yeah, I've had, thankfully not a lot of experience, but I've had at least one similar experience with that stands out in my brain of like, so it wasn't exactly like that of like the final being exponentially more difficult, but like, in my opinion, obviously, this is my, I might have been wrong in my assessment, but when I was taking virology, okay, and this professor emphasized like multiple times of like, I'm only there was a textbook for the course, and she was like, I'm only going to cover, like, I'm only going to test on like what I cover in the lectures, so like, ignore the textbook. And so I did that, and then like when I got to the exam, and maybe I just wasn't adequately prepared or not, but I'm like, but I remember like reading the exam, and like this was, and I even went afterwards, like, where in the lecture is this covered? And like it wasn't covered, but it was in the textbook, since obviously the textbook is more thorough, but it was like, it was like, I just felt like I was misled, where like she just said like, I only focus on the lectures, I'm only going to test from the lectures, and then at least in my opinion, like, she didn't end up doing that, but thankfully that was my only experience with that. And the rest, every time that a professor has said that, they have been true to their word. But that was like that one time that sticks out. That's pretty much tattooed in my brain at this point, because I wasn't actually traumatized, but I was really like, I walked in like I walked in quite confident into that exam of like, I know these lectures, and then like, it didn't like, thankfully didn't like go off the rails. So like I didn't like bomb the exam, but I'm looking to perform like exceptionally well, and I did like slightly above average or like at average, which is like just fine. It didn't end up like hurting me or anything, but there was definitely some disappointment there to be had.
Well, that was
Uh,
me too. Like I lucked out. At least I've been quite good at math in the past, so it's like I can, you know, reverse figure it out when you have the equations and everything. But
Absolutely.
yeah, the majority of the class basically borderline failed.
Yeah, I'm sorry to hear that, but I definitely know. I've heard of many complaints that happen from some students.
Yeah,
Let's see. I'm just bringing out some potential final exams. Oh, okay, great. Oh, I don't need that. Uh,
that's pretty simple. That first
uh,
one.
yeah. This doesn't seem too bad.
Yeah,
Uh,
I hope that's on the test.
it says potential final exam. There. Okay, so you're given three.
Yeah,
Oh, okay.
fatty
Well,
acid biosynthesis. What is the
uh,
committed step? Biosynthesis. So that's um
well, we can.
building.
Yeah, that is absolutely building. Which I'll bring up. This
I
is
guess you can see it right now.
for building. I don't think I brought up the building one. A fatty acid biosynthesis. 1 second. Let me bring
Yeah,
up
because
the.
we were in oxidation.
Yeah, exactly. Okay. Yeah, we are right there. Okay. Whoopsies. Uh, well, uh, shown. So this is fatty acid synthesis.
Yeah.
And well, all you should need to do is like look at the arrows in terms of going in that direction of condensation and also like another there's also another dead giveaway, not just the arrows, as in if I remove the arrows, obviously I'm not going to do that, but if I were to remove the arrows, just based off of like what you're seeing is being produced, it should be like a dead, hopefully a dead giveaway of the committed step.
Well, half of it wants me to say it's the NADPH, but then I'm thinking the CO2 loss.
Yes. The CO2 loss would also that should be a dead giveaway.
Yeah.
That yeah, because it's not a trivial matter in terms of because yeah, it's a gas and like it's just going to like it's just going to float away and like
Yeah.
and as you and so it's not there, and like also to go in reverse, it's not a trivial thing to go in reverse in terms of so actually this is actually a good thing to cover, going back
Yeah.
to so that that pyruvate carboxylase, okay,
Yep.
where it looks like it's using CO2. So that's in gluconeogenesis. Just give me a second, bring it up. Do-do-do. So I'll
But
bring up.
as you said, it was technically bicarbonate.
Yeah, so this is a little bit misleading. Well, no, I mean, it's not intentional. uh, uh, where yeah, if you just look at this, it looks like oh, like this is not this is using CO2. So it's yeah, sorry, here.
Yeah,
Yeah,
there
the
you go.
pyruvate carboxylate. Yeah, it's it's hydrocarbonate. Where yeah, we are carboxylating, but we are not using CO2 to carboxylate. It's in the
Or
form
is
of yeah.
that carbonic acid? Is that what that's technically called?
Well, yeah, carbonic or carboxylate should be the same thing.
Carboxylate, yeah.
Yes, exactly. And so and this is irreversible, and it's for that reason as well, in terms of like it's not strictly speaking CO2 that's being used,
Right.
as in like I said, anything that's like releasing CO2 is almost all, at least in humans, is going to be like irreversible, because it's not a trivial thing to carboxylate something. So what was that? Fatty acid synthesis and oxidation. So let me go back to it.
Yep.
We were right here. So all that to say is like that should be the committed step, given the fact that, well, one, the arrow's in one direction and you are releasing CO2, as in just the condensation reaction alone. And so you do need, it looks like you do need to know the enzyme for that step, which we will go
I guess
back.
I'll make sure I know to memorize the enzymes.
At least I can say that I was tasked with memorizing every single step, and then every single enzyme that catalyzed that step. Pretty much similar to what I was tasked with glycolysis.
So that should just be... Oh, okay, that's not the fatty acids. That's the beta keto acyl ACP synthase.
So that's where you'll have your fatty acid synthase coming into play. Just 1 second. Let me bring it up. You have your condensation. So, well, actually, you can almost sort of, not cheat, but like, I mean, technically the enzyme would be like fatty acid synthase, but you can say that for like every step in the pathway, as in it's just fatty acid synthase, but because like that's a obviously I don't do that. That's obviously not what they're looking for. But like,
and you know what's funny? I swear she had a example of like what not to put on the test, like, you know, those like silly examples, like they're escaping me now.
That's okay, but I'm pretty sure I have a sense of what you mean.
You know, like those memes that you see, like, you know, it'll be like a sixth grade kid that'll say something like I need to find one now.
That's okay, but some smart ass like basically answer of like,
exactly,
technically true, but clearly not the answer we're looking for. Just as like, I think this is also a nice example of like, if they ask you for what enzyme carries out this step in fatty acid synthesis, like it's technically true at some level. Like fatty acid synthase like does it all, because the course itself, as you cover, like fatty acid synthase is this large macromolecular multi-domain enzyme, so it's doing all the things. But they mean the thioesterase domain, the ketosynthase, the enol reductase, blah blah blah.
She had examples like where was the American Declaration of Independence signed? At the bottom.
That's a good example. Yeah, technically true.
Or how do you change centimeters to meters? You take out centi.
You just erase the C. Yeah, exactly.
Yeah.
They're looking for a good faith interpretation, but but there's also I would say the onus is on the professor. Like, let's just say, if I
because I mean you could almost get half marks for that technically.
I'd have no problem saying, like, if I didn't know this, but I knew fatty acid synthase, I would put that down. If they marked it wrong, like, I would have for sure contested it. I was like, I'm not wrong. You need to be more specific, or you need to like reword your question of like what domain or what is the name of the domain that carries out this reaction. But either way, onwards and upward. Let's see what the rest of the questions are. Name one hormone that regulates this enzyme and state if it is a possible or if it's a positive or negative regulator. Well, we just went through. It'd be like.
I mean, I know it was like glycogen synthase, but probably insulin.
Insulin at some level, yeah. So, well, we can also go back to a little bit where here, right? There's some regulation going on here. Not necessarily, so not for necessarily synthase, okay?
Yeah.
Because, you know, we're releasing fatty acids. But you can
Mhm.
probably also imagine, maybe in terms of, well, if glucagon, okay, is, let's say, promoting beta oxidation, okay,
Yeah.
then probably, okay, probably glucagon is, you know, doing the opposite to fatty acid synthase. As in, if glucagon, like, because like
Right.
we're not gonna make fatty acids as we're releasing them, right? It wouldn't make any
Yeah.
sense.
Exactly.
Uh, just like as we saw with like you know all the things that do to one pathway, you know, do the reciprocal or the reverse on the other pathway.
Yeah.
Typically,
So you
all
could
the
say like glucagon negatively regulates it.
yeah, in all likelihood, glucagon would be a negative regulator. Yes, and
Yeah.
and also, you know, you can even work in of what you just said. Well, if glucagon, okay, going with this, if glucagon is stimulating the release of fatty acids, okay, and thus
Yeah.
beta oxidation, well, then insulin is probably the opposite, because insulin is
Yeah.
the opposite of glucagon, right?
Hmm.
And so probably also opposing effects as well, in terms of, well, and it also makes sense metabolically, right? Of like, well, what do we if we're releasing insulin, okay, and we are under high blood sugar conditions, okay, well, what does that mean? We pretty much have we have an excess of glucose, okay, so
Yeah.
we want to store it, and if we have an excess of glucose, you know, we don't want to burn all the glucose all at once and produce excess energy for no reason, and so also similarly, okay, we're probably not going to promote beta oxidation with insulin. Like we have a bunch of glucose, and well, this also makes also makes metabolic sense in reality of like, well, what typically happens of like if someone eats, you know, kilograms and kilograms of glucose, they're going to store some of it as fat, and so you're going to be promoting or activating fatty acid synthase.
Yeah.
Where if you eat 3,000 calories of glucose or sugar, like no matter what, yeah, unless you're some sort of, you know, athlete or, you know, you're burning that much calories, which is possible, uh, three or
Well,
four
I
thousand
do
cal.
find it fascinating when they do the like um I guess overfeeding studies where they'll have like some of those outliers where they burn as much, if not more, of the calories that they consume.
Yeah, no, there's lots of interesting studies in terms of fasting and overfeeding and refeeding. So yeah, these questions don't seem so. So I think what we'll do, I think we'll finish off the fatty acids and then just like dive right into the questions, since like
Yeah.
the signaling cascade is well, we just covered it, and it's,
Relatively fresh.
yeah,
Yeah.
relatively fresh, and like you've also seen most of it before, at least 80, maybe
Hmm.
90%.
Yeah.
And so I think diving
Even
right
like
into,
the membrane stuff.
yeah, exactly, like the lipid rafts and phospholipids, all that stuff.
Yeah.
I think that's pretty much in the, you know, fresh in your mind,
Hmm.
and still stuff you've seen before multiple
Definitely.
times. So we were around where we were exactly here. Yeah. Okay. Yeah, the mobilization of fatty acids. Well, we've seen this before. Of yeah, going back to that glucagon signaling. Okay, and then yeah, your lipid drop. Well, you have your perilipin. Your perilipin gets phosphorylated. Then you have your series of events. You know, your CGI, and that results in the release of your triacylglycerol that then goes into the bloodstream, and then it gets bound to serum albumin, and then, you know, throughout the body we go. Yes, cytoplasm and mitochondria. Okay, we went through this. The carnitine shuttle. Yeah, beta oxidation, 2 carbons at a time. Okay. And I think they mentioned explicitly how many ATPs are produced from, let's say, you know, 16. Okay, yeah. You probably do need to take note of
Yeah.
that in terms of 108 total ATP from 16 carbons. Yeah.
Right.
Okay, yeah, and they go through degradation again. This is all review, but yeah, you definitely looks like you need to memorize the steps and the enzymes. Even though again it's annoying, but yeah, that trans delta 2 enoyl CoA, beta ketoacyl CoA, and for sure like the overall main steps of oxidation, hydration, oxidation, thioesterase, or cleavage. Same thing.
Yeah.
Okay, and what happens? So yeah, going through an odd number of carbons since we are oxidizing 2 carbons at a time. Well, what happens when there's 3 carbons or an odd number of carbon atoms? So yes, there's that little reaction here in terms of essentially converting to succinyl-CoA that can get shuttled into, let's say, 1/3 of the way through. It basically does not get shuttled into the beginning of the citric acid cycle. It gets shuttled into 1/3 of the way through.
Yeah.
But yes, you have a separate pathway that will essentially convert it of we pretty much, as they say there, we're going to beta oxidate 2 carbons at a time until we can't anymore. And then we're going to convert it into succinyl-CoA, and then off to the citric acid cycle we go, or the Krebs cycle. Same thing. Yeah, pretty much once we're down to 3 carbons. So, if we're 15 carbons, we go down until we oxidize 12 out of the 15, 2 at a time. So we undergo 6 rounds. Then we have 3 left. And then the propanyl-CoA is converted to succinyl-CoA and then shuttled to the citric acid cycle. Okay, and the cis double bond. Yes, how to deal with that. So just as before, we do beta oxidation until we can't anymore. We encounter that double bond. And well, here they're showing the double bond at the 9 position. And so they're just saying that it'll proceed for 3 rounds until you reach the double bond at the 9 position. And then, do they go through? Yes. You'll have an enzyme that basically isomerases, where you convert the cis double bond to a trans double bond. And then, yes, you can beta oxidate. Let me just double check. Yes, cis double bond to a trans double bond.
But it still makes less.
Yes.
ATP.
Yeah. Exactly. As they say.
Oh yeah, one fewer FADH.
Exactly. Yeah. And so therefore, you have less FADH2 getting to the electron transport chain, and thus a little bit less ATP. Yes. Great. Right. Then, well, yeah, this trans delta 2 fatty acid is that substrate for that enoyl-CoA hydratase. But ultimately, I guess your main message is your cis double bond is converted to a trans double bond, and then that can pretty much proceed as normal for beta oxidation with this enoyl-CoA hydratase. Okay. Yes, but then instead of 1 double bond, what about 2 double bonds? Which ultimately, I would say nothing dramatically different, other than, yes, your beta oxidation occurs as normal until you hit your first double bond. And then, yes, you're converted to trans, and then that trans follows the same pathway as well before, in terms of it's a substrate for enoyl-CoA hydratase. And then, yes, this beta oxidation then completes normally, yielding cis delta 4 fatty acid, which is, right here at the end of the pathway. So, starting with 2 double bonds, okay, we isomerase to a trans double bond. And then we pretty much have our beta oxidation completed, and then we end up as a cis delta 4 fatty acid.
So maybe slightly off topic, but
that's okay.
In the scope of this, why is it that trans fats are so bad technically?
Yeah, so that's a great question. So it's actually pretty depicted nicely here. So it's not that they're. I mean, they are, let's say, bad in this, but they're bad because
Yeah.
they largely behave like saturated fatty acids. Where how do saturated fatty acids behave? Well, so, and again, saturated fatty acids, they're saturated with hydrogen, right? And so
Yeah.
you have your long hydrocarbon tails, okay?
Right.
And you have your long straight hydrocarbon tails, right? Just at well, bringing it back to the membrane and phospholipids in the membrane of like your unsaturated phospholipids. Okay, you have that little kink in your fatty acid that
Yep.
makes that pretty much introduces a gap. Okay, and so trans fats are typically bad. Okay, as they're shown here, where this is a trans double bond here at that position, where even though it's a double bond, comparing the double bond on the left to the double bond on the right. Okay, the double bond on the left, you have that kink there, like you see with that
Yep.
kink in the phospholipids that have a double bond, as opposed to with the trans. It pretty much looks like a normal fatty acid. Like if you compare it to the end, it's just like your typical conformation, right? Despite the fact that there's a double bond. So this is essentially mimicking a mimicking a saturated fatty acid. And if it's mimicking
Right.
a saturated fatty acid, there is more, and this also overlaps nicely with the membrane and phospholipids of like when there's no kink in the double bond, okay, and then you're not introducing that gap, okay, then your fatty acids they can stack together nicely and form these nice hydrophobic interactions. Okay, and if they can form these nice hydrophobic interactions, well, then they can essentially aggregate much more effectively and efficiently. As in,
Right.
let's say your saturated fatty acid, like butter, right?
Yeah.
Butter at room temperature is solid, right?
Yeah.
And it's because as a result of you have these saturated fatty acids that are forming these interactions, and therefore you can basically form a solid like butter, as opposed to like olive oil or like your unsaturated fatty acids that are typically liquid. Okay, and because they are not, they cannot pack as nicely with their hydrocarbon tails, since those would be in your typical fatty acid, like let's say olive oil. They would be in a cis double bond. Okay, and so they're kinked, and so they can't pack together nicely, and that's why unsaturated fatty acids, like olive oil, they tend to be liquid. They basically can't pack together nicely like a saturated fatty acid. So for trans fats, it's essentially they're mimicking a saturated fatty acid, as in they have the double bond, but since they're not kinked, they can pack together nicely and basically behave like butter, and thus like clog your arteries like butter, so to speak.
Right. And so they're basically much more dense per volume, plus
Yes.
the fact that you don't actually get as much energy out of them as well.
Yeah, that's true. You would not get as much energy out of them. That's true. But like, yes, you're absolutely right. That's the perfect way of putting it. They are more dense because they can pack together nicely, because they can form those nice packed hydrophobic interactions between them. Just
Okay.
like you see with the membrane, in terms of when you have a kink in your phospholipid in your hydrocarbon tail, you have that gap there. But then, well, relating back to the membrane of like what can cholesterol do, it can fill in that gap, and basically that's why cholesterol has typically it results in more rigidity in the membrane, because you're
Right.
with the cholesterol you're plugging that gap, and so with saturated fatty acids there's no gap to plug, and they pack together nicely and they become dense, and
Right.
clog your arteries, and therefore
Okay.
trans fats, they essentially behave like saturated fatty acids, because as you can see there, it adopts the same conformation as a saturated fatty acid, where you see here at both the same, you have a cis double bond and a trans double bond, and the trans double bond looks the same as a single double bond there, of just
Right.
nice whatever triangle, so to speak.
Well, and obviously, it takes our bodies a lot more to break these things down.
Yes, that's I don't know about a lot more, but yes, a little bit more.
I mean, somewhat more, I guess, in the grand scale of things. Like, if you're
Definitely.
consuming a large proportion of them.
Yes, absolutely, definitely more steps for sure. But
Yeah.
the main issue with trans fats is like that, and that was the whole, because you might remember of like, when they first arrived on the market, it's like, oh, these are great, so like these are better, right?
Yeah.
And like at a molecular level, like there's some element of truth to that, not really, but like you can make the case when we didn't know any better of like, oh, we have a double bond here, okay, a double bond like we see in unsaturated fatty acids,
Yeah.
but it's clearly not the same double bond, and that's why it's like, oh, turns out they're actually not better for you, and we can't make the case, and then it also makes sense at a molecular level because these, generally speaking, the trans fats were made by like basically like trying to add hydrogen to these unsaturated fatty acids, but like
Right.
they were not able to, or at least originally, when they added the hydrogens, like they adopted the trans conformation, and not the cis. But
Right.
yes, in a nutshell, that's why trans fats are basically the same as saturated fats.
Yeah.
But they, at one point, they gave the illusion that they were better for you because they had that double bond.
Right.
Okay, two double bonds continued. So, but once we get to this cis delta 4, okay, then we can deal with that, where we essentially turn it trans again, okay, where you have your acyl CoA dehydrogenase that is introducing another double bond, but then eventually, okay, it all becomes trans, and you get a trans double bond, and then this can be oxidized into acetyl CoA. But basically, your main message is like when you have multiple double bonds, they are essentially converted into trans double bonds, and then those can be beta oxidized, essentially normally. But yeah, you have a whole bunch of rearrangements occurring when you have two double bonds. Okay, yeah, ketone bodies. Which, yes, what happens when there's no real fatty acids to burn, and no glucose, no carbohydrates to burn either. And then this is where the ketone bodies come into play. And again, as maybe a review, like ketones, it's that C double bond O with two carbons to the side. And yes, these are an alternative fuel for the brain, where, like I said, the brain prefers glucose, but when no other options exist, it can run on ketone bodies
Yep.
in your typical healthy human. And so yes, how are your ketone bodies formed? So yes, your main site is in the liver, and one of your primary ketones is acetone. Okay, where you go from acetoacetate to acetone. That's sort of like that's like your base, your most fundamental ketone body.
Yep.
That can result in your acetone breath, as they say.
Mm-hmm.
Okay. Let's see. So this is the formation of ketone bodies. I assume and hope you don't need to know the full pathway. Otherwise, I would say that's a lot. If you're tasked with knowing both the pathways for fatty acid synthase and beta oxidation, and it's fair because these are important pathways. I would say, hopefully, you don't need to also memorize ketone body anabolism and all those.
Yeah.
Which I don't think you do.
In the practice questions, but
Yes, we will. Yes, ketone body catabolism. No surprise, we can break them down pretty much going in the reverse. And yes, as in ketone bodies, we start with acetyl-CoA. We are condensing acetyl-CoA to produce our acetone and our beta-hydroxybutyrate. And then your catabolism, we're essentially going in reverse. And we are ending up with two acetyl-CoAs. which then can be shuttled into the citric acid cycle, and then for energy production. Okay, yes, fatty acid biosynthesis. Okay, where, yes, this is biosynthetic, and so yes, it is using NADPH, since, as I try to emphasize, NADPH is typically reserved for biosynthetic processes, although it's not exclusive. And yes, all we're doing is essentially building up 2 carbons at a time, our triacyl glycerol. And yes, starting with acetyl-CoA, we're going to see converted to malonyl-CoA, and then round and round we go in fatty acid synthase. Okay. Acetyl group shuttle. Let's see. Yes. We have... Just trying to... Like, there's nothing...
I think this was supposed to be just kind of like a broad...
yes, that's what it looks like. So...
so I'm just gonna hit the bathroom again real quick.
Yeah, go for it.
It's ready to go.
Alrighty. So yeah, there is a point to this figure.
Okay.
The point is, yes, to get acetyl-CoA, okay, to the outside, because fatty acid synthase, okay, occurs not in the mitochondria, okay, as in it occurs outside the mitochondria. But understandably, we have a lot of acetyl-CoA in the mitochondria, right? What do we do? How do we get our acetyl-CoA out? As in, if we're going to carry out fatty acid synthesis, and we need a supply of acetyl-CoA, which we do, well, we need to get our acetyl-CoA out of the mitochondria. And yes, this is the process of getting your acetyl-CoA outside of the mitochondria, and they're going through the reactions here, where it's pretty much, yeah, this cycle here. Well, acetyl-CoA multiple sources, such as whatever before the citric acid cycle, or from glycolysis to pyruvate to acetyl-CoA. And then, yes, citrate is basically your mediator, okay, where just as with the malate dehydrogenase, okay, that was the way of getting NADH outside of the mitochondria. It's a similar principle and concept of like, well, acetyl-CoA is not directly transported outside of the mitochondria. We basically have a middleman in the form of citrate. where citrate, or rather citrate synthase, acetyl-CoA, catalyzed by citrate synthase, we get citrate. That is transport outside of the mitochondria, and then, okay, citrate lyase converts it back into acetyl-CoA, and then that can be shuttled to fatty acid synthesis. That is the whole point of this process here. And so, yes, that's essentially the way of getting acetyl-CoA outside of the mitochondria so we can carry out fatty acid synthesis.
Okay.
Yes, that definitely makes more sense. So, yes, this is our
so in a way, it's like citrate is a acetyl-CoA shuttle.
Yeah, you could think of that way. Like obviously it's not directly like carrying acetyl-CoA outside, but it's like, yeah, it's like whatever. I'm going to be converted. I can pass through the membrane, and then I'll be converted to acetyl-CoA once I get outside of the mitochondria. But yes, citrate is essentially our acetyl-CoA transporter, so to speak. Okay, yes. Malonyl-CoA is our sort of like basic building block of fatty acids. Where yes, the malonyl-CoA will be made from acetyl-CoA. We're basically going to carboxylate it using hydrocarbonate or carbonic acid, essentially the same thing. And yes, we have our acetyl-CoA carboxylase. Okay, so getting into our acetyl-CoA carboxylase. That yes contains biotin, okay, important vitamin or micronutrient, and so we're going to have, okay, this is similar to the acyl carrier protein in fatty acid synthase. We have a biotin carrier protein here with the acetyl-CoA carboxylase, as in, so here, that's why, so what's happening is we are carboxylating our acetyl-CoA to generate malonyl-CoA. And this is a nice example of like we're carboxylating, but we are not carboxylating with CO2 directly, as in it's starting off as hydrocarbonate here, HCO3 minus, okay, and that gets bound, okay, so here, like it's now it's CO2, but it's not in gas form, like it's basically it's been captured, as in starting off as yes, exactly. Where it's not, this is not extracting like a gas, from the environment. It's like extracting hydrocarbonate, and then yes, it's what gets bound is the CO2, and then that will be condensed with acetyl-CoA, now generating malonyl-CoA. Where yes, we just like with the acyl carrier protein in fatty acid synthase that has this arm that swings from domain to domain. It's a little bit simpler here we have our biotin carrier protein that's swinging from the carboxylase domain to the trans-carboxylase domain, and then it condenses with acetyl-CoA to generate your malonyl-CoA there on the right-hand side. So yeah, pretty much your main steps is hydrocarbonate or HCO3 comes in, and then CO2 gets bound, okay, to your carboxylase domain. Your acetyl-CoA and your CO2, your bound CO2 condense, and then that generates your malonyl-CoA, which will then go on to fatty acid synthase.
Right.
Okay. So carbon atoms are added 2 at a time by a repeating 4-step sequence. I'm curious. 1 second. Okay, no, never mind. Okay, yes. Our major steps here, condensation, reduction, dehydration, reduction. But a little bit more nuance. we have a keto reduction and an enoyl reduction, but still a reduction just the same. But it matters whether whether we are dealing with a ketone or an enoyl intermediate.
Right. I think I'll
but
try to commit that to memory just in case.
Yeah, the more the better. Definitely. You almost certainly need to commit the domains of fatty acid synthase to memory. And like, yeah, there is a keto reductase, yeah, shown here. You have your KR for keto reductase. You have your ER for enoyl reductase. You have your DH for dehydrogenation. And yes, you have your malonyl acetyl-CoA ACP transferase for MAT. And yes, your keto acyl ACP synthase. But yeah. Okay. But at least based off of this.
Matt is definitely a lot easier to say.
Yes, 1000%. But yes, 2 carbons at a time, using NADPH, because this is a biosynthetic reaction. Yes, the growing fatty acid chain is covalently attached to a small protein. Yes, the acyl carrier protein, or ACP for short. All right. So, yes, fatty acid synthase gets charged with acetyl-CoA and malonyl-CoA. So basically, we're going to be carrying out, our condensation, okay, and pretty much smashing together, sort of, acetyl-CoA and malonyl-CoA. And then, yes, CO2 is released, which we've seen before. Like, yeah, that first step. This should be your committed step.
Yeah.
Given the fact that, yes, we are releasing CO2, and they're also depicting it as a line going in one direction. So that should be absolutely your committed step.
Technically, all of them have one direction, or one, as if you're going by arrows.
Yes, if I remember correctly. Like, it is all one arrows. I think that's why in the question, I think they specify, well, they say the committed step, as in, like, if you're committed, like, well, the first one is going to be the committed step, generally speaking.
Step 1.
Yes.
Starting is the committed step.
Yes, and there's no turning back now. Which also, let's say, makes intuitive sense of, like, if you're consuming this, because we're condensing malonyl-CoA and acetyl-CoA. Like, we don't want to waste that. Of, like, if we're going to use acetyl-CoA, like, why turn back? Or,
well, and the entire synthase enzyme is kind of already bound.
Yes, yeah, exactly.
So it's not going to be unbound until it's cleaved.
Exactly right. So, but yes, we've linked together 2 carbons each, generating 4 carbons, but we started with malonyl, and that's why that decarboxylation step is important, of malonyl-CoA 3 carbons, and then during the process we lose CO2, then we've linked together 2 carbon, or two carbon units that are linked together on the acyl carrier protein, or ACP. And then, yes, we get a reduction. No surprise, NADPH is playing a role there. And yes, you have your beta keto acyl reductase, or the keto reductase for short, the KR. And then, yes, you get your dehydration, or your beta hydroxy acyl ACP dehydratase. Probably need to know that, given the fact that this is in red, and it looks like all of these are in red. You probably do need to know, and like, yeah, you're typically tasked, just like I was, to memorize all of the enzymes and all of the steps of beta oxidation and fatty acid synthase.
Yeah.
But yes, get dehydration, and then yes, you get another reduction, enoyl ACP reductase, where now, again, the ultimate endpoint, so to speak, at least for this carbon chain, is we want to get to hydrocarbons, just a bunch of reduced carbons. So no surprise, we have 2 reductive steps. And then, once we elongate to the appropriate length, okay, 16 carbons, 14 carbons, 12 carbons, then your last step is that thioesterase to release your fatty acid. Otherwise, it's just going to be stuck there. Okay, and yes, they go through the structure. Yeah, this large macromolecular protein, multi-domain protein, with, yes, your various domains, ketosynthase, your dehydration, enoyl reductase, ketoyl reductase, and your thioesterase domain. And yes, it's conserved throughout many organisms in the tree of life: plants, bacteria, vertebrates, fungi, etc., etc. I think this is just more like a factoid. I don't think you really need to know this. Yes, multi-enzyme. So that's why, I don't know, it's a little bit of a weird question to word that way. Going back to, like, yeah, name the enzyme that mediates the step.
Yeah.
But whatever. Especially when they're saying it that it's a multi-enzyme complex, but whatever, I'm not going to argue.
Well, it might be a gimme.
But definitely, you know, the more you memorize this, definitely the better. 6 enzymes, functions, plus ACP in one. Well, yeah, if it's one polypeptide chain, you know, this is like, you know, it's all the same.
Yeah.
So it's fatty acid synthase. You can just say with the enzymes, fatty acids, technically speaking.
Yep.
All right, initiating fatty acid steps. Is this a review of what we just went through right now? They're just going into more detail. 1 second.
Think so.
Pretty sure this is the exact same thing.
Yeah, and I think it just
yeah, initiating. Yeah,
I think it was because it was just trying to say that, like, you know, it goes through multiple rounds.
Oh, okay, yeah, that makes sense.
Until it hits the 16.
Yes, but yeah, this is one round, and yeah, it's many steps, but pretty much like, yeah, your acyl carrier protein essentially moving from domain to domain. It's depicted nicely here. Yes, you are condensing here, and then your acyl carrier protein, okay, goes to the keto reductase domain. Then you get a little bit reduced, and then it should swing over to, yes, your dehydration domain. You lose a water molecule, 2 hydrogens, 1 oxygen. And then, yes, now you're back at, no, not back. You are at your enoyl reductase domain. You get reduced again, reduction of double bond. And then now you are at essentially at least fully reduced for 1 round. But, you know, round and round we will go. Many, many times. Yes, condensation is just going through all the same things. Okay, enoyl reduction. Yeah, and given that it's in bold, you know, I had to memorize all these things here. The trans delta 2 butanoyl ACP.
Yep.
Not fun. Yeah, you can transfer butyl group from ACP itself. This is repeating all the same steps. Okay, well, they're just going through the synthesis of palmitate, 16 carbons, which is going to be multiple rounds. Like 8 rounds-ish.
Yep.
Yes, and yes, the substrate is physically linked to fatty acid synthase through the whole process. That's why you have at the end your thioesterase domain that pretty much cleaves it off and releases the fatty acid. As they're saying there, that is highlighted, the thioesterase domain. Okay, well, this is review, literally what we just went through. Okay, yeah, okay. This is the assembly of triacylglycerol. So, yeah, review. Longer chain fatty acids. Well, palmitate, 16 carbons, but we also have longer ones like linoleate, 18 carbons. 18-2, which should be two double bonds at the 9 position, at the 12 position. Yes, 2 carbons at a time. Nothing remarkable here. But yes, well, what if we want to actually produce an unsaturated fatty acids, as in, we saw how to deal with a double bond, but what if we want to actually introduce a double bond? Where yes, now we can actually make cis double bonds. But yes, it only at we can't introduce a double bond anywhere, only at the 9 position. And let's see, yes, which must be reproduced using NADPH. So yes, you essentially have your fatty acyl CoA desaturase, not the most clever name, but thankfully it makes sense. But if we are, all they're saying here is like this is coupled to something, and it's coupled to the essentially the oxidation of NADPH to NADP plus, where yes, when so this is, let me just double check if this is an oxidation. Yeah, that all makes sense. Where to introduce a double bond, we are carrying out this reaction that's then ultimately coupled to the oxidation of NADPH, producing NADP plus. Okay. Desaturase requires oxygen, which must be re-reduced using NADPH. Yeah. Okay. Yeah. Gotcha. Where to basically to like recycle this, as in like this cannot carry out like in perpetuity, or basically it can't do it alone, as in like basically there's a modification that happens that the NADPH has to come along to basically reset the fatty acyl CoA desaturase. Yeah, the fatty acyl CoA requires oxygen, which must be re-reduced using NADPH, as in like, yeah, you basically need that NADPH for the fatty acyl CoA desaturase to be able to do its thing. They basically require, or rather the NADPH is required to make that double bond. And yes, and aside from that delta 9 or that double bond at that 9 position, we pretty much, as they say, we can't introduce any more double bond. And so if you want 2 double bonded fatty acids, then we basically need to we need to consume them in some form or another, or consume like some sort of precursor to have our polyunsaturated fatty acids.
Right.
Yes, an essential dietary fatty acid. Okay. So yeah, we went through fatty acid synthesis, but technically we actually haven't fully assembled like how we actually store our fatty acids, which is in triacylglycerols. But not overly complicated in terms of like all we're essentially doing is linking our fatty acids, our long chain fatty acids, 16, 18 carbons, to our glycerol scaffolding. And we have potentially multiple sources of glycerol, as in you can have your dihydroxyacetone phosphate, okay, pretty much converted to glycerol 3 phosphate, or rather to glycerol 3 phosphate right down here. And then also, yes, your glycerol itself can be converted to glycerol 3 phosphate. But basically, we're going to we have multiple sources of glycerol, and that glycerol is going to be activated. It's activated by pretty much phosphorylating it, so no surprise there's a kinase involved there. And then we're essentially going to start linking our fatty acids to our glycerol molecule, or basically 3 fatty acid chains to each carbon on our glycerol, where essentially, yes, we end up with our triacylglycerol at the end. Let's Just making sure I'm extracting everything. Yeah, you have acyl CoA, but this is just basically like multiple rounds. So then starting with glycerol 3 phosphate. Okay, we're just linking our fatty acid chains to it, to all 3 of the carbons. Okay. Yeah, no, not a surprise. Triacylglycerol can be made in adipose tissue or liver. Okay. Let me just see. Triacylglycerol can be made in adipose tissue or liver. It is simultaneously broken down only in adipose tissue. Approximately 70% of fatty acids released by lipolysis, so basically the breakdown of fatty acids, are reesterified to form triacylglycerols. Oh, I see. Right. Yeah, this is all like in sort of in balance or like in flux of like, well,
Right.
Basically everything's active like all at once, so to speak. In terms of like we're always undergoing beta oxidation and like, you know, doing the reverse, but basically, you know, obviously it's more nuanced than that, but basically to some baseline level, okay, as they say, there's broken down and we're making essentially at the same time, where as they're broken down, some of them are reesterified to form triacylglycerol. So as we're releasing fatty acids, well, then the reverse is also happening to some extent. Like, well, then we're making our fatty acids as well, or our triacylglycerols. And so what they're saying here, like when they say this futile cycle, it is the analogy would be of like we are using glycolysis and gluconeogenesis. It's like, well, the futile cycle for that is glycolysis and gluconeogenesis occurring at the same time, which doesn't happen. Okay, but now here it's different. Of like at some baseline level, both pathways are operating at the same time. To some, obviously not both of them at 100%, but some baseline level. Like when we release fatty acids, some of them are basically reformed back into triacylglycerols, and as they're saying, like there's probably a reason for this, yeah, in terms of like, well, yeah, to essentially meet metabolic demands, which is we always have some sort of supply ready to go for whatever purpose that we need, and we don't need to, like, you know, basically we don't need to do any dramatic shifts, so to speak, of like, well, this pathway was totally off and now we need to turn it on. It's always on at some baseline level, so we just need to ramp it up, and that's easier than just like going from fully off to on.
Right. And hence why people with you know, metabolic disease typically have high triglycerides in their blood.
Yes, that is true too. Absolutely. That's a fantastic connection to make as well. Where, yeah, you're putting it into the larger context. Okay, and yes, so regulation of fatty acid synthase or oxidation. No surprise, some similar themes here. Although, okay, yeah. But yes, you have your rapid response, slow, moderate, and gradual adaptive. Yes, and hopefully most of these are intuitive. Of like an allosteric regulator, it's going to be like more immediate, or definitely way more rapid than hormonal, where you need some sort of signaling cascade, binding of your hormone, your ligand to your receptor, and then going through all the steps. And then yes, changes in gene expression. You know, obviously. Not much longer, but relatively speaking, longer than you know, an allosteric regulator, because you have to not only activate the respective gene, you need to undergo transcription, you need to undergo translation, and then potentially get the protein to where it needs to go in the cell or in the organism. Oh, interesting. They're sort of okay. I would say they're kind of tricking you. The transfer of fatty acids into mitochondria is the committed step. Okay, fair enough. Uh, I don't know that.
Um, I mean, this is under regulation of fatty acid beta oxidation, not biosynthesis.
Uh, yes. Okay. Yeah. I was just thinking, making sure. I want to see
Yeah, I
that
know it's like
clearly.
so close.
Uh, okay. Uh, even then, like I sort of take issue with when they, yeah, I guess, but they're saying like the transport or something across the membrane is the committed step. We don't really typically refer to a committed step that way, but I take the point. Like that is
Yeah.
true. Uh, that like yes, if we're transporting fatty acids in the mitochondria, we're going to be breaking them down. But still, even then, we're say this is an atypical use of committed step in biochemistry. But either way,
Yeah.
I'll go definitely follow what's here on this slide here for beta oxidation. If they consider that the committed step, then who am I to argue? Yeah, once it's in the mitochondria, as in transported by carnitine, or let's say acyl carnitine, and the transporter devoted to getting your acyl carnitine across the membrane inside the mitochondria, that is your committed step. Where yeah, once you're in the mitochondria, like you ain't going back out, basically.
Yeah.
Yes. Okay. Regulation. Uh, so yes. Your insulin is whoopsies. So, well, citrate lyase, as they're showing here. So they're showing here that insulin activates citrate lyase. Now, does that make metabolic sense? Well, citrate lyase catalyzes the conversion from citrate to acetyl CoA, and then yes, acetyl CoA can be condensed into malonyl CoA, where this, if you recall, of for fatty acid synthesis that occurs outside the mitochondria. So we need to get our acetyl CoA outside the mitochondria, and
Right.
citrate is that key player in getting
Yeah.
acetyl CoA outside the mitochondria. And so, like, does it make sense that insulin would activate this? Well, yes, it makes metabolic sense. Yeah, in terms of under if you're releasing insulin, then you have high blood glucose. Okay, and that's essentially energy surplus conditions, right? And so if you're generally speaking under energy surplus conditions, you're going to be making fatty acids, and so this would stimulate, or make sense that this would stimulate overall fatty acid biosynthesis of insulin. And then glucagon essentially ultimately leading to the reverse. More specifically, as they're showing here, glucagon is inhibiting acetyl CoA carboxylase. Okay, and that acetyl CoA carboxylase, yeah, well, you are essentially you are inhibiting fatty acid synthesis, which also makes sense and all the other things here of glucagon, epinephrine. Glucagon, you're under energy limiting conditions, and that's why you want to mobilize your glucose stores. But then also at the same time, you want to mobilize your fatty acids potentially, which we saw, right? We saw with the original signaling pathway of glucagon binding to its receptor, and then you undergo all of those steps to phosphorylate your perilipin, and that releases your fatty acids from your lipid droplet and then into the bloodstream. Sort of one of those first slides. And so all of this makes sense metabolically. And also epinephrine, where again, under fight or flight, you want to mobilize your energy stores. So it makes sense that you would not undergo fatty acid synthesis when you are fighting or fleeing from something.
I wonder, does epinephrine trigger lipolysis as well?
As in beta, essentially fatty acid breakdown.
Yeah.
Almost certainly, yes. As
Yeah.
in virtually 100% certain. Yes.
Like, so it would function the same, say, like that first slide where it showed glucagon. it would function similarly to, phosphorylate perilipin,
Well,
leading to the cascade of events,
yes.
releasing a monoacylglycerol.
Yes. Yeah, exactly. A monoglyceride.
Yeah.
Uh, but it's not necessarily the case that epinephrine will bind to like this and like not necessarily this unique pathway of adenylyl cyclase cyclic AMP. But it will be like
Yeah.
something. It will be something in common here of like yes, perilipin being phosphorylated. You have your CGI and then your ATGL that results in the
Yeah.
release of your triacylglycerol, and then yes, your monoacylglycerol fatty acids to serum albumin in the bloodstream. Yeah, absolutely.
Interesting. Yeah.
Oh yeah, we were right.
Probably just not as strongly, I'm assuming, considering, most stimulant fat burners are not really going to do too much for fat burning per se.
Well, yeah, just as you're showing here, of glucagon playing a multiple role in terms of and also here you're seeing reciprocal roles of glucagon, right? As in, we literally just went through how glucagon results in the release of fatty acids, right?
Yeah.
And then here it's like inhibiting, it's playing a dual role and also inhibiting fatty acid biosynthesis.
Right.
And so epinephrine, like, yeah, you are almost with I'm like virtually absolutely certain that it will stimulate fatty acid oxidation, breaking down.
Yeah.
But probably almost certainly more strongly, it's like pretty much breaking down glucose as in mobilizing those energy stores, because like even though you can burn glucose and fatty acids for energy, like you can burn glucose way faster than
yeah.
fatty acids, and so you would prefer to burn that.
Yeah.
But almost certainly, like you're almost certainly going to mobilize both. So like whatever it is, mobilize glucose, mobilize fatty acids, and like again, if you're in fight or flight mode, it's just like, well, whatever happens, but you're probably going to preferentially burn glucose because it's way more efficient. Okay, yes, regulation of acetyl-CoA carboxylase. Yes, this is your key regulator of fatty acids synthesis. So, which is going back to that biotin carrier, protein. The acetyl-CoA carboxylase that catalyzes the synthesis of malonyl-CoA, where you're pretty much carboxylating acetyl-CoA, and then you generate malonyl-CoA, and then that will condense with another acetyl-CoA.
Right.
Okay. So that step that generates your malonyl-CoA, that is understandably regulated. So epinephrine glucagon causes phosphorylation, inhibiting it. Okay, which again makes sense in terms of if we are under fight or flight, or we're under low energy conditions, such as when glucagon is released, or low energy conditions stimulating the release of glucagon, we don't want to synthesize fatty acids. And then yes, as they're showing it here, insulin promotes the dephosphorylation of acetyl-CoA carboxylase that will promotes the synthesis of fatty acids. Regulated by metabolites, so yes, citrate, palmitoyl-CoA, which makes sense. Definitely the citrate making sense in terms of the citric acid cycle, and pretty much going back to energy charge.
Yeah.
Okay. Yes. fatty acid degradation are regulated so only one occurs at a time, although even this is sort of like contradicting what it said earlier in terms of but this is probably not a strict binary. But let's zoom in a little bit more. Okay, so high blood glucose results in insulin release, so high insulin. And then yes, you have your ACC here. So this is just going through the steps that we just went through of that's your acetyl-CoA carboxylase. Okay, and it is active. It is active when it's dephosphorylated. Okay,
right.
And so what's occurring? Pretty much what we just went through. In the text, it's like with insulin release. Okay, that means that we have high blood sugar, okay, and that we are under energy surplus. So if we're under energy surplus, then we want to favor fatty acid synthesis. And so how do we favor fatty acid synthesis? Well, the active form of acetyl-CoA carboxylase, the ACC, the active form is dephosphorylated. So it makes sense that insulin is stimulating the phosphatase or activating the phosphatase, favoring the dephosphorylated form of acetyl-CoA carboxylase that plays a critical role, pretty much one of the first steps in fatty acid synthesis. So far, all of this makes metabolic sense. And then, glucagon. You're favoring the phosphorylated state of acetyl-CoA carboxylase, as in you want to be inactive because under low energy conditions, you don't want to be making fatty acids. So you want your acetyl-CoA carboxylase phosphorylated and thus inactive. And so it makes sense that the glucagon is activating the protein kinase A, which the protein kinase A will phosphorylate your acetyl-CoA carboxylase. So all of this is tracking metabolically, and we've already went like once you have your malonyl CoA, then they're just showing the different fates in terms of fatty acids. Transported to the mitochondria to make acetyl-CoA, or fatty acids, or malonyl CoA to make fatty acids. That's pretty much your sort of mediating step.
Yeah.
Or your branching off point. Okay. Yes, the tissues, your stereotypical textbook figure. Okay, which you probably know this from your physiology, I would imagine. Okay, yes. Liver. Definitely covered this before in terms of, yes, major site of storage of glycogen and therefore glycogen synthesis. And yes, releasing glucose from glycogen or gluconeogenesis when levels are too low. And yes, there's also this aspect of ketone bodies as an alternative fuel, when necessary. When you've essentially depleted all your carb stores, you're not consuming any more carbs afterwards. And yes, hepatocytes, your fancy terminology for liver cells, they will upregulate or downregulate, whichever the appropriate genes to express the appropriate enzymes to meet the metabolic demands of the organism. Where yes, no surprise, let's say if you're under super carb-restricted conditions, you are going to upregulate the enzymes devoted to making ketone bodies.
Yeah.
Nothing surprising here. Yes, adipose tissue convert glucose to fatty acids, which now we can totally make the link in terms of, how glucose, how an excess glucose can promote the formation of fatty acid synthesis and fatty acid storage, because we know the metabolic pathway under which glucose can be converted to fatty acids, where pretty much glycolysis to pyruvate, pyruvate to acetyl-CoA, and that acetyl-CoA can be used to make fatty acids. Okay, yes, and no surprise, they respond to hormones such as epinephrine and insulin, which we just went through. Where epinephrine, you want to mobilize your carbohydrate stores, release them into the bloodstream. You also want to mobilize your fatty acid stores. So epinephrine, you're not going to be favoring fatty acid synthesis. And the reverse for insulin, where again, for insulin, you're under essentially energy surplus, like high blood sugar, so you're going to be favoring fatty acid synthesis. Okay. Glucose 6-phosphate in the liver. Yes, the multiple. Yeah, nice path. Nice figure to show the multiple fates for glucose 6-phosphate. Where yes, it can be converted back into glucose, back into the blood, or glucose 6-phosphate shuttled into the pentose phosphate pathway to make ultimately NADPH and the precursors for nucleotides, specifically your ribose 5-phosphate. And then yes, your glucose 6-phosphate, obviously, for glycolysis down to pyruvate. But then the pyruvate can be converted to acetyl-CoA that has multiple fates of acetyl-CoA to the citric acid cycle, obviously, but then the acetyl-CoA to make fatty acids. So this also shows a nice link between, glucose and fatty acids. And also a nice link between the glucose 6-phosphate and liver glycogen to make glycogen, but to reinforce the point or the concept, it's like, well, would glucose 6-phosphate be directly used to make liver glycogen? I guess that would be like the question.
Would it be?
Yes, as in like to make glycogen.
It would be converted into like glucose and then UDP glucose.
So yes, so yeah, but there's that.
Actually, yeah, it'd be glucose 1-phosphate, then
yes,
UDP glucose.
Perfect.
Yes.
Yeah, exactly.
Glucomutase.
Yes, that phosphoglucomutase.
Phosphoglucomutase. Yeah, there we go.
Okay, yeah, metabolism. This is just like a nice summary. I don't think there's anything new here that we haven't seen before. No. Okay, yeah, hormonal regulation. Well, this just overlaps with everything that we just covered with signal.
Yeah, kind of similar to what you were talking about too.
Yeah, exactly. Okay, yes, the well-fed state, or yeah, the energy surplus, where no surprise, you're gonna upregulate glycogen synthesis, and yes, the glycolysis, pyruvate entry into the citric acid cycle, all the things, fatty acid biosynthesis, the whole problem, so to speak, with the well-fed state, energy surplus, making too many fatty acids, typically. Yes.
The chronic state most of our society lives in.
Yeah, absolutely. I mean, this is all stuff that, like, I know that you know of insulin response and lowering glucose levels. And yeah, getting glucose transporters to the membrane. Triacylglycerols. This is pretty much just like wrapping up and summarizing everything. Let's see. Yeah, epinephrine, impending activity, flight or fight. Yes, acts on muscle, liver, adipose tissue. And yes, we've seen it before. Epinephrine makes sense, like I said. And just shown nicely here, of epinephrine should also stimulate the release of fatty acids. We want to mobilize all our energy stores, our glucose from our glycogen, our fatty acids from our lipid droplets, or our triacylglycerols stored on our lipid droplets. All of this, is making metabolic sense. And yes, in muscle, increases fructose 2,6-bisphosphate. And well, we went through how fructose 2,6-bisphosphate plays a role in regulating glycolysis, as in, the phosphofructokinase 1, and whether in the presence or absence of fructose 2,6-bisphosphate, that will alter how PFK1, its percentage activity, either stimulate it or inhibit it, whether fructose 2,6-bisphosphate they're saying here that in the presence of fructose 2,6-bisphosphate, that will activate phosphofructokinase 1, and therefore you are stimulating glycolysis. And then, yes, all the reverse is true. Insulin versus glucagon, where, yes, if glucagon is being released, then we're not in energy surplus. So if we're not in energy surplus, well, we're not going to be synthesizing fatty acids. We're not going to be undergoing glycolysis, okay, which makes sense since there's no glucose there to burn. And yes, gluconeogenesis to supply other tissues. All of this is making metabolic sense, and yes, the release of fatty acids, of glucagon under low energy conditions. Then yes, we want to mobilize our fatty acids and release them. Okay. Well, it's just going through all the signaling between liver, brain.
Well, it's kind of cool because it's like a visual depiction of what you've kind of asked me a couple times.
I guess so. Because we are an organism. We are an integrated system between brain, liver, muscle, all that stuff, and highly coordinated. Yes, no more glycogen, so glucose must be made from amino acids. During prolonged fasting, but that's a separate topic that maybe you'll cover in a future biochemistry course. But this is important too, in terms of transamination, and all that stuff. Okay, the stress state. This is all just going through the different metabolic states. Let's see. Steroid hormone, including low blood glucose, release of fatty acids. Although I don't know that it's a strict binary. Well, you when we cover this, I remember you made the nice distinction between chronic versus acute stress. Right? And that might have, you could, as in fight or flight is also a stress, right? And so for that, you want to release your fatty acids and release your glucose into the blood. But here you're promoting gluconeogenesis and the storage of glycogen, where a stress state can also be fasting, as well. So this is a little bit more gray, and not super binary. Okay, diabetes. I know that you know this in terms of basically, unregulated blood sugar, either whatever type 1, which is essentially genetic, like insulin deficiency, the body's not making insulin, or type 2 insulin sensitivity, or insulin insensitivity. The body is no longer responding to insulin the way it's typically supposed to be. And so yes, without an insulin response, metabolism is dictated by glucagon. And yes, it is not stored or used as a fuel. And despite the fact that glucose is present, but it's present in the blood, it's not getting inside the cell. So basically the cells and the organism is not getting the signal that you're actually well fed. And so yes, it thinks it's fasting. Okay. Yes. And you're not going to be undergoing glycolysis. Yes, excess blood sugar stored in the urine, covered that before. All this is pretty much just summarizing everything that we just went through. I think maybe, okay. Well, we've covered GSK 3. I think this is all just review. I think I want to dive into the questions. Start. Yeah. Oh, well, I guess this is important too. The regulation of GSK3. Wow, where, yes, so rewinding back to glycogen metabolism. Okay, so we have glycogen synthase. Okay, and yes, glycogen synthase is inhibited by phosphorylation. And there's multiple things that can phosphorylate glycogen synthase, but one of your key players is this glycogen synthase kinase 3 that inhibits it strongly. Okay.
Yeah.
So, well, then, but how is the glycogen synthase kinase 3 regulated? 1 second. Well, not a surprise. It's essentially auto-regulated, so to speak. But they're showing here how GSK regulates glycogen synthase. 1 second.
Come on.
So yes, you have pretty much what they're showing here is your interaction between glycogen synthase kinase 3, okay, and glycogen synthase, okay, and that, as they're saying here, the GSK3 can only phosphorylate a serine if the residue for C-terminal to it is a phosphoserine, as in, like, pretty much it can only, you basically need, like, this docking site, okay?
So it has to be like a phosphorylated serine?
Yes, as in what they're saying here. So the residue 4 C-terminal, whoopsies, scrolling around a little bit too much. 1 second. Where if that serine at position 4 there, the +4, if it's not phosphorylated, then essentially, like, your GSK3 can't dock, okay, and therefore it cannot phosphorylate your glycogen synthase. So that's what they're saying here in terms of, like, the priming site phosphorylated by casein kinase 2. So basically, in order for GSK to phosphorylate glycogen synthase, well, glycogen synthase already needs to be phosphorylated. It needs to be phosphorylated at that position. And then at that position, at that serine, that's 4 residues away. As in, let me find my cursor right here. So going back to, let's say, put it into whoopsies, the larger context of recall that you have your glycogen synthase kinase 3 here, okay, and I guess they're naming it based off the fact that there are 3 phosphorylation sites on glycogen synthase, okay,
right,
and so in order for glycogen synthase kinase 3 to phosphorylate, okay, glycogen synthase, it already needs to be phosphorylated once, okay? And so what they're showing here, what those 3 plus signs represent, it's like, well, pretty much like those 3 phosphorylation sites, and they show specifically in this diagram these 3 phosphorylation sites here, as in your 3 serines, 1, 2, 3. Those are your 3 phosphorylation sites. What they're saying here is, like, for the glycogen synthase kinase 3 to be in the right position to phosphorylate glycogen synthase on its 3 sites, it needs essentially sort of like this docking site, or it needs to be primed of, like, well, at that serine on glycogen synthase, that essentially forms the docking site for the glycogen synthase kinase 3 that can then go on to phosphorylate glycogen synthase on those 3 serine residues.
Right.
That's, in a nutshell, a big nutshell, what's going on. Where, yeah, your glycogen synthase already needs to be phosphorylated once at that specific position, so that your GSK3 can then come along and then do its 3 phosphorylation events afterwards. Okay, yes, and then now they're showing here can only phosphorylate serine if the residue, no, what I just said.
Yep.
Well, but yeah, then they're showing here of like the subsequent, then you get now everything phosphorylated at the serines.
Right.
But the original one on the right, that was already phosphorylated. That was carried out by another kinase, as in when they say that priming site phosphorylate, yeah, that casein kinase 2 phosphorylated at that serine, and then that allowed the GSK3 to bind and dock and then carry out its phosphorylation events. Okay, but then, you know, how do we turn off, or how do we ensure that GSK3 is off? So, pretty much, well, it can get phosphorylated on its own serine. Okay, as in it's going to be phosphorylated by PKB, and that effectively blocks the docking site. As in, instead of being docked on the phosphorylated serine on the glycogen synthase up here, it's basically docked to itself. Yeah, just have that phosphate blocking or occupied there, and then when it wants to. Well, with glycogen synthase, it would be docked at that serine that's phosphorylated on glycogen synthase. Okay. No, and the rest is just review. Yeah, everything that went through fatty acid synthesis. Okay, great. All right, we can jump into the questions.
I'm not sure too much how primed my brain's gonna be at this point.
Yeah.
A bit burnt out now.
No, yeah, no, I would say maybe just like a couple of questions. I'll just pull.
Yeah, for sure.
Yeah. Well, so since hopefully it's a little bit fresh in your mind, which of the following best describes the effects of insulin signaling on glycogen synthase kinase 3 and glucose transporters in liver cells? So like this is a fairly tricky question, because you gotta think through overall like the metabolism of like, okay, GSK3, okay, that glycogen synthase kinase 3, that's responsible for phosphorylating a glycogen synthase, okay, and when glycogen synthase is phosphorylated, it is inactive. Right. As in like,
so we would want to activate GSK3.
Right. So glycogen synthase, yes, that as in under what conditions do we activate glycogen synthase, at least in the context of the question? Well, we would actually activate it, okay, when glycogen synthase is dephosphorylated. Okay, so it's a lot of metabolic logic. Okay, and so well, when is glycogen synthase going to be dephosphorylated? Well, it's definitely not going to be dephosphorylated when glycogen synthase kinase 3 is active. As in, if glycogen synthase kinase 3 is active, as we just saw and went through, it's going to phosphorylate glycogen synthase and inhibit it. Okay? Right. So, what do we want to do at the very least? We want to inactivate GSK3, as in like you should be able to eliminate D and E, right? Okay. As in, does it make sense? Like, do we want to activate GSK3? If we activate it, then that will go on and phosphorylate. Well, I need to double check. Yeah. Sorry.
It'll phosphorylate glycogen synthase.
Yes, it'll phosphorylate glycogen synthase, which is what we don't want, because we want active glycogen synthase, because we have excess glucose, right? As in, again,
so we want C technically.
Yes, exactly. where we want to be able to take up glucose from the blood, and then start promoting glycogen synthesis. So
Yep.
yes, we want to inactivate the kinase so that the kinase does not inactivate glycogen synthase, because we want glycogen synthase to do its job. And its job is to make glycogen under high blood glucose conditions, which we are.
Right.
Okay, yes.
So in my idea about how glycogen synthase was working was right in my mind, but I was flipping insulin and glucagon around for some reason.
I mean, these are all confused. Like even it's sometimes definitely even difficult for me. Of like, yeah, insulin, glucagon, all the hormones, and just with the names, like it gets super confusing. Okay. When applied to isolated kidney tissue, I'm not even sure I've ever heard of this.
Aubane? Aubane?
Inhibits O2 consumption by 40%. This means that in kidney, potassium O2 exchange is inhibited by. Well, they're changing up the spelling, but whatever. Regardless, O2 exchange is active by O2 uptake. Actually, I'm not even sure about this question. When applied to isolated kidney tissue.
I guess so. It's disrupting the gradient.
Yes, absolutely. A toxin that blocks the sodium-potassium pump inhibits O2 consumption by 40%. This means that in kidney, potassium O2 exchange is inhibited by potassium O2 exchange is activated by. Well, at least my intuition at least leads to B, you're probably not activating that. And you're probably not blocking. Well, yeah, if you're blocking O2 consumption, then you would not be inhibiting O2 consumption.
Yeah.
Where you'd be inhibiting, like blocking is like typically inhibiting by 100%.
Yeah.
As in, I personally would I would eliminate B and E. But this is a tricky question, actually.
I guess if it's saying that there is such a thing as a potassium oxygen exchanger or a sodium oxygen exchanger, it would just be so like you would need, if it's inhibited by, so I'd assume inhibits consumption of O2. I'd assume because sodium is typically being pumped out, that would rule out C. If it's inhibiting the consumption. Because then it's not being pumped into the cell,
yes,
right?
Yes. Okay. Okay. Sorry. Yes. The sodium potassium ATPase. Okay. So for blocking that, yes, I agree with your analysis. As in, I think
it should rule out C, D, and E. So then it would be A, probably.
I'm not so sure about. So I'm on the same page. I'm on the same page as you in terms of eliminating C. That makes sense to me.
Yeah.
And B and E, at least my intuition leads towards that. I am a little bit conflicted in terms of A and D.
Yeah, I just don't understand. Well, I guess it's, maybe. Well, the ATP shouldn't have anything to do with consuming oxygen, technically.
I agree with you, but I think approximately 40% of ATP is consumed by. Oh, okay. When applied to isolated condition, it blocks that. The sodium potassium ATPase. inhibits. I almost want to. 1 second. Huh.
Definitely nothing in the slides. We just went over.
Unless this is just like a strict, sort of experimental question, in terms of like, well, just, follow the logic, which is, that's why I said, like, you can probably just eliminate the one that's like you're just blocking. Well, if you're inhibiting, you're not blocking. So this is our sodium, potassium, ATPase. It's right here. Okay, so what are they saying in the question? 1 second. They are saying inhibits O2 consumption by 40%. This means that in kidney. Okay, I mean, I'm not even sure that, like, I think this is just trying to, whatever, get you to overthink of what's the relevance of kidney. You can just think of, like, yeah, any cell that has a sodium potassium pump or ATPase. So yes, E out, B, C, and out. Now, approximately what is NV2 is consumed by the, so yeah, we have it here. I am trying to think. It's inhibited. Oh, okay. So a potassium O2 exchanger. I think sort of a trick question, maybe.
Yeah, I guess because like there shouldn't be anything else inhibited. It says it specifically says it's a toxin that blocks the sodium potassium ATPase. So that should rule out A, B, and C, right?
I agree with you. Yes. I really think the only thing like whatever I don't think we covered any potassium O2 exchanger like at all.
Yeah,
I'm pretty sure that doesn't even like really. I'm not sure if it exists. I've never come across it. As in like just by like E like it's out for sure. I think just by process of elimination by D alone. Okay. No, I can't highlight that. But that's a really tricky question. I would almost say like almost an unfair question.
What question is that? Yeah, it is D.
Okay, great.
Yep.
Then, thankfully.
Well, process of elimination. But it's definitely a tricky question. I guess it's meant to be like a tricky question.
That is true. That definitely makes you, yes. But this doesn't make me think biochemically. Like I would have just again just based off experience of like whatever you're talking about inhibition. Well then we're not talking about blocking, and so I just like whatever that's out for me. And then if you're talking about inhibition, then we're probably not talking about activation. And so that's why I just put B and E out, and then the rest is like well. I don't recall anything about potassium O2 exchangers. Okay, so what's
I thought it was supposed to be like maybe a conceptual question. It's like, well, if there is such a thing as a sodium oxygen antiporter or something, like
that could hypothetically exist, but this is just this is like a real curveball.
Yeah, like I don't think there's such a thing, but
But definitely that is a tricky question. But we got it. Okay, but it definitely took some time, for sure.
That's one of those ones that I'd skip and come back to.
Even me too, for sure. Actually, but okay, the metabolic function of the pentose phosphate pathway is like we covered this like directly.
Yeah, which should be B.
I agree with you. Generate and pentoses for the yes, biosynthetic fatty acids and nucleic acids.
Yeah.
Participant oxide. Yeah, let's double check if anything's right. Synthesized phosphorus pentoxide. No. Provide intermediates for the citric acid. Definitely not that. Okay. Yeah, this is a good example. Transport of fatty acid from the cytoplasm to mitochondrial matrix requires well, there's definitely a couple of them that you can eliminate right off the bat. That's for sure.
Yeah, anything that doesn't have carnitine.
Yes.
Uh,
And I would say there's another one that it involves carnitine, but there's something there in one of those
yeah,
answers.
hexokinase.
Yeah, exactly. Right.
Um, I is it? I don't remember coenzyme A ever being mentioned. So
Uh, uh, so maybe not sort of super explicitly, but the, through let's say process of elimination, like so I don't necessarily expect you to recall, but if let's just say if you remember what this does, pyruvate dehydrogenase, okay, then that should give you a clue as to why like this should stand out as like an incorrect answer.
is that to? Oh yeah, that's to break it down into acetyl CoA.
Yeah, exactly. The pyruvate dehydrogenase complex, pyruvate
Yeah,
to acetyl CoA,
yeah,
and so like yeah, this is like this is all they're doing is
yeah.
changing two of the three are the same, and then so understandably, it might not be super clear that how coenzyme A is involved. But just by virtue of hopefully recalling that the pyruvate dehydrogenase complex,
Yeah,
plays a role in the conversion, well, plays the role in the conversion of acetyl pyruvate to acetyl CoA.
yeah.
Great. And this should be a question like right up your alley because we like this is something that I drilled into your mind multiple times.
Oh yeah, okay. So
So well, so they're giving you the enzymes, but you should
yeah,
be but
um
you should be able to at least get the beginning and the end, and then
yeah, thiolase is the end, and the
or no, sorry,
hydratase, or
yeah,
no,
yeah,
yeah,
yes.
it'd be the, is it, mm,
So we can go like go with what you know, right? Like, you know the thiolase is at the end, right? So there are definitely some answers here that you can eliminate right off the bat, right?
yeah, um and hydratase is in the middle. So okay, yeah, so thiolase is at the end. So yeah, obviously eliminate A and E, and then hydratase should be the second, so it should be C.
Let's. All never hurts to double check. My
Yeah.
intuition tends to agree with you. Yeah, the acyl CoA dehydrogenase, and well, yeah, and well, like you just went through the analysis. The only one that makes sense based off what you know of thiolase at the end and the dehydration. Sorry, the hydration at the second step.
Yeah.
Only C fits that criteria, so that should be the answer. And
Yeah.
4 and 1, like yeah, if you remember the acyl CoA dehydrogenase as the beginning, and the beta hydroxy acyl CoA dehydrogenase
Yeah.
as like a third, then you're in better shape. But yeah, thankfully,
But
just
hence why I said I should probably commit the actual names to mind, just in case.
yes, no, for sure. But
I mean, it
thankfully,
is nice that I was able to,
exactly.
determine it.
Which is great. This is I would say this is a fair multiple choice question.
Yeah.
Where if you know the names of the enzymes in the order, like easy question. But if you just have
Hmm.
a little bit of like the fundamentals of like the order of the reactions and the type of reactions,
Yeah.
oxidation is at the beginning, but it's not clear like is the acyl CoA dehydrogenase my oxidation, is my beta hydroxy my oxidation. But go with what you know. Like
Well,
you
I
did a
already
quick.
figured out the easiest way to, remember it is just, A comes before B.
Yeah, whatever memory aid works. Great. Uh, okay, ketone bodies are formed in the liver. Maybe just do like yeah, a couple more questions, and then like you probably need like break, sleep, food,
Yeah.
all that stuff. I understand it's long.
All of the above.
Uh, but maybe simulating stress conditions of an exam.
I forgot that
Uh,
it's used mainly as um is the
Yeah, so
Is it
this I think this
acetone?
is a tricky question.
Yeah.
Where so but there are some that you should be able to eliminate right off the bat, hopefully.
Yeah. Well,
Uh,
not lactic acid.
I agree with you. Is there
Um,
any other? I would say at least one probably that hopefully you can eliminate right off the bat.
acetoacetyl CoA.
Yes, since
Yeah.
that's essentially like like basically the first step of formation of ketone bodies, of just like condensing two acetyl CoAs together to generate
Yeah.
acetate. But yes, it's the rest is pretty tricky. Okay. And the liver and transport into the extrahepatic tissues.
Like I remember acetone is the like one of the final byproducts.
Yes, is like the pretty much like your primary ketone body. Uh, but if you recall, I don't necessarily expect you to. I'm gonna close these. I have a little bit too many tabs open. Uh, where so for ketone bodies, like yeah, you have your acetone as like one of your main ones, but then you do generate another ketone body, a lesser known ketone body, which it's go back to it, where they're sort of
I
like
just I mean obviously it's beta hydroxybutyric acid, um, but is it it probably would have the CoA.
did I skip it?
Um,
I'm just going through ketone bodies, or trying to find ketone bodies, but maybe I skipped it. Uh,
I see ketone bodies
I see it
metabolism.
here. Okay.
Yeah, that's what I found.
I guess it's further. Oh yeah, here it is. Okay, yes.
Yeah,
Uh,
so
yes.
beta hydroxybutyrate, but
So yeah, so it should be the other as in so not the acetone,
Hmm.
as in
but I
it
think there
should be.
was two beta hydroxybutyrates, wasn't there?
I think one is the CoA and the butyr.
Oh,
So
butyryl CoA.
yes,
Okay, so it should be the butyric acid.
yes, butyrate and butyr not, technically not the same thing, but they're used interchangeably. Just like aspartate and aspartic acid, although they are technically different, but we tend to use them interchangeably.
Right. Okay.
Uh, uh, but yeah, this is a little bit of a tricky one. But
Yeah.
you can there's definitely some that you can eliminate right off the bat. Lactic acid,
Yeah.
acetyl CoA, and
I'll be
maybe
honest. I probably would have thought acetone just because.
which is understandable because like that is like the main that well one like I did emphasize that is like the main ketone body, right, and like that results in your
Yeah.
acetone breath.
One, and
But
and that's why I kind of like I was thinking like maybe that was
so
you know
the not the trick, but the so going back to the lecture slide where okay, what's the point of let's say ketone bodies? The point well, it's like yeah, we're under like very carb restricted conditions and maybe starvation conditions. And do they show it? Yes. Where uh yeah, it's shown here of your acetoacetate and your D-hydroxybutyrate exporters energy for source for heart, skeletal muscle, kidney, and brain.
okay.
They
Yeah.
want you to like memorize or know that little small detail. But like yeah, acetone is like
I,
your
I guess it never explicitly said D beta hydroxybutyrate acid.
no, you're right.
It was
Uh,
just implied, I guess.
Uh, it so it's a little bit kind of maybe a little bit unfair, but it is technically speaking, or like if you answered let's say CoA, and like the answer is butyric acid, well, the answer is butyric acid, but like you could potentially contest and say like, well, it is and the butyrate version is not the same as the butyric acid, and so you can totally say that you were confused and you just put down that one as the as your answer, but
right.
Colloquially speaking, okay, like we very often use the ATE form of a molecule when we're talking about, so we can talk about aspartic acid, right? Glutamic acid. But then there's aspartate and glutamate. And technically the difference is it's like glutamic acid. That's when you have your proton, you're in your acidic form, and then when you're glutamate, you've lost your proton, and then now you're like negatively charged. And so strictly speaking, this beta hydroxybutyric acid is not the same as your butyrate, but sort of like colloquially, like no one gives anybody a hard time of like if you refer to either aspartate or aspartic acid. Let's put it that way, unfortunately, or like they're used interchangeably, unfortunately. But yeah, it is a little bit strange that they're using different terminology than what is used in the slides. Let's say like that's a little bit unfair.
Well, it's possible that like this could be an old test that was based off a old set of slides that actually said that more explicitly. I don't know.
Fair, but they should hopefully give you appropriate things to study from. Okay, and yes, so I think maybe just 9 and 10, and then I'll stop the torture. But we definitely covered a hydropathy plot, right?
Yes.
And they should hope that I'd say there's definitely quite a few that you can like eliminate as like incorrect answers.
I think it was E.
Let's
because it was that was to determine the membrane spanning segments.
So are there ones that, let's say, you're confident that they are not the correct answer, as in like we know that these are incorrect?
I'm just trying to see. Yeah, I'm nearly certain that it was E because that was the only plot that
no
we looked at.
I agree.
I remember.
I was just like, I agree with you in terms of what. I was just curious if like other ones stood out of like, you know that there are, as in like pretty much you're confident that all the other ones are wrong, right?
Yeah.
Okay. Yeah. Great. I agree with you. But some might be tempted to like maybe.
Yeah, I mean that would make intuitive sense, but it's just I explicitly like I don't remember it being called a hydropathy plot, so that's a good reminder. But I remember there was like one specific plot that we went over.
Yes, exactly. Great. I'm like, that easy question, which is great.
Yeah.
And well, the specificity of the potassium channel for K plus over, I don't necessarily expect you to remember, but totally like we touched upon this. I do remember that, but there are definitely some.
The differential interaction.
Yeah, you're almost. I haven't even read the rest. Hydrophobicity of the channel, phospholipid composition of the channel. Well, does that even like? Could you even explain like why this is like? Yeah, I agree. Like, and so why doesn't it make sense? Are you able to like formulate like the phospholipid composition of the channel? How does that? I agree with it doesn't make any sense.
Because it's I mean channels are made of amino acids.
Yeah, perfect, beautiful. As in if you just remember like your basic biochemistry, like we're talking about a channel protein, right?
Yeah.
And it's a protein made of amino acids. So like, well, probably not the presence of carbohydrates in the channel. Presence of cholesterol in the channel, that would be more like in the membrane and other parts. Like, yeah, we've definitely, I don't think we've covered anything, and you almost will certainly never cover anything like cholesterol inside of a protein, or only
No.
under most of the niche conditions.
Yeah. And there's
Uh,
carbohydrates like maybe, and not even on a channel, I don't think. Is anything like that?
yeah, within a channel, no, we have definitely covered carbohydrates like modifying a protein, right? And like
Yeah.
glycoproteins and all that stuff. But yeah, totally.
And like I would potentially think the hydrophobicity of the channel, but it's like uh it was more the selectivity filter that it had to do with for potassium over sodium, even though
Yes.
like uh you know the difference in size is pretty massive. It was the specific interactions.
Yeah, perfect. So, and so if if you just like obviously, if you remember straight from like the slide and like what we covered, like yeah,
Yeah.
that is your right answer. But if you're stuck, usually my sort of rule of thumb, like if I'm stuck on like whatever a question is, like go back to basics, and like if you
Yeah.
hopefully remember like fundamental biochem of like yeah, if we're talking about a protein, well then it makes no sense to have a phospholipid inside of a protein,
Yeah.
especially the channel. Makes no sense to have a carbohydrate inside the channel of a protein. Definitely never
Or
spoke about.
cholesterol.
But for this one, okay, you can eliminate this one through the fundamentals of biochemistry, where okay, uh, if you relate it to the plasma membrane of like if you happen to recall maybe a little bit of good review of like if we're talking about a plasma membrane, a phospholipid membrane, what are things that can typically pass through a phospholipid membrane, and what are things that typically cannot pass through
Uh,
a phospholipid membrane?
hydrophobic molecules.
Uh, yes, hydrophobic molecules can pass through a membrane because the
Yeah,
phospholipid membrane are hydrophobic. Okay.
exactly.
And what are the things that pretty much it's like a non-starter? You cannot pass through the membrane.
Yeah, anything hydrophilic.
Yes, anything, and hydrophilic would be anything polar and
Yeah,
charged. Okay,
yeah,
and
exactly.
so it makes no fundamental sense if we're talking about charged things, something like potassium, and we're talking about hydrophobicity of like uh, the hydrophobicity of the channel allows potassium to pass through. That would be like potassium passing through the membrane, which it doesn't at all. It
Yeah,
can't.
I
Uh,
mean, I guess like the way that I would potentially try to interpret that is like, you know, if it's talking about how little hydrophobicity there is of the channel,
uh, yeah,
like because it doesn't really emphasize if it's like being very hydrophobic or
but even then, like given that's fair. But given the fact that it's charged, like even like the hint, the hint of hydrophobicity, is just like a no, I don't expect you to know that, but like yeah,
yeah.
the fact that it's charged, like any sort of like if you're just like over 2%, 10% hydrophobic, it's just like not gonna happen. Just
Mhm.
because you're pretty much we're talking about something that's charged, it is so on the sort of like opposite end of hydrophobicity that is just like never going to happen. But yeah, like
Yeah,
you got this from straight knowledge, which is great. I think overall, like you're
we can go over 11 too.
sure, yeah. Which of the following is not involved in the specificity of the of signal transduction? Interactions between receptor and signal molecules, location of receptor molecules, structure of signal molecules, transmitting transporter signal molecules by receptor molecules. Okay. Um.
That's a bit
Well.
of a tougher one.
Well. Yeah. It's like pretty much. It should be like four are correct and one are wrong, so to speak. Uh,
Yeah,
the way that the question is framed, right?
yeah.
Uh, and so, which of the following is not involved? Well, or you can frame it another way of like, which of these is involved or are involved in specificity, and which ones are not? Okay.
Yeah.
And so, are the interactions between receptor and signal molecules specific? Would you say yes or no to that? Wait,
Yes.
I would agree with you in terms of like, yeah, go
Oh, I
back
guess
to
location isn't really
uh,
specific.
uh my intuition tends
Well,
to like agree, like it can get really nitty gritty, like, but I would definitely keep that as like this is
yeah.
potentially a right answer. I would retain this as like a potential right
Mhm.
answer if there's absolutely no better ones here.
Yeah.
And then yeah, what about the structure of receptor molecules? Is that involved, or based off of
Yeah, because obviously it's not going to bind if it's not the right structure,
exactly? Generally speaking, we're talking about protein.
which would go the same for the signal molecules as well.
Exactly. As in, like, when we're talking about any type of ligand receptor of like
Hmm.
insulin binds to the insulin receptor and other ones too, but it doesn't bind to just any random receptor, right?
Exactly.
It doesn't it doesn't bind to your opioid receptors. If it did, that'd be great because but it doesn't. And so yes, pretty much generally speaking, there's always exceptions, but ligand receptor interactions are specific. So just about anything to do with the ligand receptor and their interaction is going to be specific. So like yeah, A, totally part of specificity. C, totally part of specificity. D, totally part of specificity. And then what about E? Transmembrane transport of signal molecules by receptor molecules. Okay, interesting. That's worded not the greatest.
Yeah, weird.
Transmembrane. Oh, okay, okay, yes.
Like, would that be like transporting the signal molecule through the?
Yes. As in like pretty much like uh well, I guess yeah, transporting the signal. So transmembrane transport of signal molecules by receptor molecules, where basically, let's try to bring up.
Like, is it saying like transporting the signal molecule through the membrane?
That's how I. Let me see if I can. Let me bring up. The signal, some sort of signaling cascade. A full cascade. Of I sort of interpreted. That's again, it's not the best wording. Um, of let me just go back and read it. Transmembrane transport of signal molecules by receptor molecules. Okay, so I can interpret that in multiple ways, but the way I interpret it is using this figure here. It's basically when your, let's say in this case your hormone binds to your receptor. I guess this probably the GPCR is not the best example. Uh, totally covered other. I guess yeah. I guess I can use this one. Of like so when insulin binds its respective receptor, insulin receptor, okay, you get to dimerization, okay,
Right.
and then okay, it's not intuitive, okay, but and this result, this is a tyrosine kinase, okay, and we
Yep.
autophosphorylate now. Is it the case? Is it the case that let's say tyrosine kinase activity only results in like one thing of, let's say, it only responds to like insulin, and like it's only going to be one outcome like no matter what, like tyrosine kinase devoted activity is devoted to this and this only.
Uh, like creating the cascade. Yes.
You're creating the cascade, yes, but is it created? Is it? I guess
like
I,
specifically phosphorylating the
no,
like each other, I
yeah, I'll
guess,
word
in
my
or
the way I
itself.
worded my question is poor, as in, is it only insulin that results in tyrosine kinase activity?
Uh,
And that there's
no,
nothing.
it's the dimerization, or it's
You're right.
like the bringing together of the molecules
Yeah, I agree. But is it only
or the
insulin that?
receptors.
Yes, but is it only insulin that results in the dimerization? As in, we only see this with insulin and insulin alone.
With the insulin receptor?
No, no, just that's why I said with tyrosine kinase activity. Like, yes, the insulin receptor happens to be a tyrosine kinase
No,
receptor.
no, there are other receptors that are tyrosine kinase.
Uh, yeah, exactly. As in other hormones or other molecules or other ligands. Okay,
Yeah,
they result in tyrosine kinase activity and its
yeah.
respective signaling events, autophosphorylation, and your whole signal cascade. Uh, and so I think definitely E is a candidate. Okay, we basically narrowed it down between B and E. Okay,
Okay.
now location, like I would say it's totally
I,
understandable, and
I guess it could be specific because it's like if you don't have a specific receptor on a particular cell, then it's not going to bind specifically to or that cell.
yeah, exactly, and we've seen it in terms of like recept, in terms of like
Yeah,
what upregulates receptors of like you have to have changes in gene expression to like let's say get well it's not a receptor but it's a good example of like what we covered of like we only want glucose transporters there, okay, when glucose
yeah,
is present in the blood, okay, and we're not just randomly like putting glucose transporters like anywhere, and we're not randomly expressing them at random times. It's like it's in
yeah,
response to a specific signal. We upregulate the gene responsible for the glucose transporter, and then the glucose transporter goes to the membrane.
yeah. Like
Uh,
specifically insulin and GLUT4 in the muscle.
Perfect. Yeah, wonderful.
Yeah.
And
So it is.
yes, but this is worded like yeah, that takes some figuring out, or at least interpretation, or
Yeah.
translation of what exactly they're asking for. But yeah, they're pretty much asking of like the signal, the transmembrane signal of like what gets transferred between the membrane and like depending on the ligand that binds in terms of, yeah, I think the example that I gave is a nice, hopefully a nice example in
Yeah.
terms of like ultimately when a ligand binds to a receptor, you're like, yeah, we're ultimately trying to get a signal from outside the cell to inside the cell. Okay.
Yeah.
And like, yeah, is it only insulin that generates this signal inside the cell, as in tyrosine kinase activity? The answer is definitely no, as in, as I attempted to emphasize. So, like, all these signaling cascades, like, yeah, we're using examples. And I think actually, uh, one second. Just to, like, I guess, wrap up because it does tie it together. 1 second.
Yeah.
Yes, perfect. Where this is a great example of like it's
Yeah.
not only insulin, right, that
Yeah.
results
Like
in tyrosine
EGF.
kinase activity, EGF as well. And so that's what that question was driving at. Poorly worded, okay, but that is the
Yeah.
case. Because like yeah, that is I had to do some processing. What are they asking for exactly? But that is what they're asking
Yeah.
for.
Okay.
Uh,
Yeah, that makes sense.
and yeah, hopefully now you can see it in terms of like we're talking about a receptor, and you can see now the two receptors, tyrosine kinase activity. We just saw it with insulin, and we just saw it with we see it here with EGF, and so hopefully it's explicitly clear that like yeah, this is not the tyrosine kinase activity. That's not specific to insulin or EGF.
Yeah.
Multiple things. Great. So I think overall, actually, you are in good shape. I
Yeah.
think with the more
And
studying.
I'll definitely, I'll go through the rest of this before, like, thankfully, my test is pretty late in the day. So
Great.
yeah, I'll definitely make sure I cover all this multiple times before.
But I think already, like you're in good, as in like you. No, we didn't answer that. You answered. Yeah, that one, no problem, because like
Yeah.
we covered that specifically, pentose phosphate pathway, and if that's all you need to know for the pentose phosphate pathway, like easy marks.
Yeah.
You got that one perfectly. And even without again knowing the actual enzyme names, right? If you just
Yeah.
knew the fundamental, you did know the fundamentals, just the order of the reactions, the type of reactions. You only
Oh,
one.
but I know now. A before B.
Yeah, yeah, exactly, perfect. This one's a little bit tricky. Okay,
Yeah.
and you got that one perfectly. Uh, and then yeah, you got that one no problem right off the bat.
Yeah.
So I think we covered like I didn't do the math, but I think we covered like whatever 8 or 10, and you got like
Yeah.
80%.
Perfect.
So with more studying, yeah, I think you are in great shape, and I have no doubt that you can and will do well.
Awesome.
So yeah, this has been great. I'm not going to keep you here any longer. I've already kept you here for quite a while. Well, first I want to thank you. I want to thank you for agreeing to do this. Super
Oh
appreciated.
yeah, no problem. Hey,
It's
I
been a
appreciate it too, man.
oh, good, good. I'm glad. And like it's been a while since like I've streamed anything, so this is a good return.
Yeah.
So I'm going to. So well, your exams in the afternoon or evening.
Yeah.
Then yeah.
2:30.
Okay. Then yeah, I'll probably I'll for sure reach out. Maybe not tomorrow to like give you a break and like
Yeah.
harass you right after your exam. But and also I don't expect you necessarily to like have your grades right away.
Yeah.
But like yeah, I will be following up hopefully to see how you're doing and see how everything turned out, which I'm confident it'll turn out well. Um,
Man.
and just out of curiosity, if you want to say like
Yeah.
the I assume you're taking classes next semester in the winter.
Yes. So right now I have a second neuroscience course, and biomedical, it's a pharmacology course, so I'm pretty excited for that.
Sounds cool.
Uh, and it might be another biology course. I need to double check. I'll fill you in after.
Yeah, yeah. I don't know. I was just out of curiosity.
Yeah.
But do you happen to know like if like is this it for biochemistry or like you have to take like there's another one that you.
Hopefully not. Like there's nothing that I need to take for my degree, but I enjoy it. So I mean, I want to take as much of it as I can, and I know it's going to help in like, anything medical related. So
Oh yeah, absolutely.
yeah.
And if anything, in my limited experience, and in my opinion, take it for what it's worth. Like the more biochemistry that a physician knows, I would say the better overall in terms of just
Oh,
understanding what's going
100%.
on
Yeah,
in the
exactly.
body. Because ultimately, it all breaks, not all, but it mostly all breaks down to biochemistry.
Yeah, exactly.
Where we had nice examples of cystic fibrosis, and like, well, that ultimately boils down
Yeah.
to like, unfortunately, something going on with the chlorine transport and chlorine channels,
Oh
uh,
yeah.
encoded by the CFTR gene.
Well, even
Uh,
just like how all I mean, our drugs work in our systems. It's like, you know, upregulating receptors or blocking them or,
Absolutely.
all of
That's
the
why
above.
I love it as well. And it also, as you know now, like it informs like everything in terms of understanding response of physical activity in the body and how to
Yep.
like optimize and also how to like hopefully filter out good versus bad information when consuming
Oh yeah.
online fitness content.
Yeah. which is 90% garbage, unfortunately.
Yes, exactly. All right. Well, yeah, it's been great. Thank you so much for agreeing. And yes, I'll let you go, eat, sleep, re-energize, take a
Yeah.
break, do whatever you have to do. And yeah, we'll be in touch. You got questions, comments, concerns?
No, could be everything, man. Really
All right.
appreciate it.
No problem. Take care. Be well. We'll be in touch.
All right. You too. Thanks.
Do. All right. That was great. Really enjoyed that. Could do that probably. Well, I wish I could do that every day. Uh, one. All right. Well, yeah, I've been streaming for a while. A return to stream. Thank you, anyone, for tuning in and passing by. Noda Bud, great to see you. Dr. Lewis, thank you so much for the questions. And Doffy, great to see you. I will be back. I should be back actually next Tuesday, getting back to my biochemistry lectures. And yeah, thank you everyone for passing by and the great feedback and questions. And yes, until next time, take care and be well.